Riscrivere il tempo. Invertire il corso dell'orologio biologico. Utopia o realtà? La prospettiva di raggiungere una terza e quarta età in piena forma e salute e di prolungare la vita ai 120 anni richiede comportamenti consapevoli fin da giovani. 70 tra i più autorevoli scienziati al mondo si riuniscono a Milano per parlare delle nuove sfide della longevità. Venite al Milan Longevity Summit dal 14 al 27 marzo a conoscere i grandi guru della longevità e imparare i segreti di una lunga vita in salute. L'evento è organizzato da Brain Circle Italia in collaborazione con le eccellenze milanesi e con il sostegno del Comune di Milano e della Regione Lombardia. Al Milan Longevity Summit si parla non solo di ricerca scientifica, ma anche di intelligenza artificiale, di lavoro e di organizzazione della società e delle città per fronteggiare il cambiamento demografico. Per consultare il programma, prenotare un posto o accedere alla diretta streaming, visita il sito milanlongevitysummit.org. L'ingresso è aperto a tutti e gratuito. Vi aspettiamo!
Buongiorno a tutti, penso che ormai mi conoscete, sono Viviana Casam e vi ringrazio di essere qui, grazie, ringrazio tutti di essere qui, soprattutto con questa bella giornata che uno non ha tanta voglia di chiudersi in un teatro, però vedo che siamo in tanti, so che ci sono anche molte persone che ci seguono online e oggi è una giornata particolarmente densa e interessante perché uniamo la scienza, e la scienza ad altissimo livello, a quello che è un fatto di costume, che è il fatto che tante persone, tanti miliardari, tante città stanno nascendo per la longevità. E quindi ci chiediamo come mai questo fenomeno e che cosa si aspetta la gente che addirittura spende moltissimi soldi quasi per essere un guinea pig, come si dice in italiano guinea pig? Una cavia, ecco, scusate, io sono 2 giorni che parlo italiano e inglese e il mio cervello non sempre fa in fretta a passare da una lingua all'altra. Va bene, allora io direi di cominciare subito. Volevo dirvi una cosa, molti mi chiedono questa bellissima immagine come è nata, è stata generata dall'intelligenza artificiale, un artista che si chiama Naima, perché sento un eco c'è qualcosa che non va col microfono, sento una forte eco, voi mi sentite bene? Sì. Ok, le abbiamo dato un input che era di creare un'immagine che non fosse né uomo né donna, perché la longevità riguarda tutti, che desse un'idea non di vecchiaia, perché parliamo di come mantenersi giovani, quindi non volevamo un vecchio, non voleva essere una maschera di qualcuno che è vecchio e giovane, e poi volevamo una cosa che desse l'idea del tempo, di tornare indietro nel tempo. Tutto questo, questi input, poi una bella immagine che ricordasse l'arte italiana, il Rinascimento, eccetera, l'intelligenza artificiale ha sviluppato questa bellissima immagine. Ecco, ve lo dico perché mi è stato chiesto da molti e pensavo che forse vi interessa saperlo. L'artista che l'ha fatto si chiama Naima e la ringrazio. Allora, grazie, questi sono per Naima, non per me. Ecco, vi ricordo poi che il Longevity Summit prosegue nei prossimi giorni. Domani sarà ancora al Franco Parenti con una sessione organizzata dalla Fondazione So Longevity e si parlerà delle città del futuro che dovranno tener conto di una popolazione che invecchia. Poi avremo una bellissima sessione il 26, una doppia sessione, una sulle reti di assistenza e il problema dell'assistenza e il contributo di pubblico e privato che sarà fatta alla Fondazione Triulza nel nuovo polo dove c'era l'Expo una volta, che adesso si chiama Human Technopole, e un'altra sessione al Teatro MIT che invece riguarderà un po' tutto il mondo intorno alla longevity, e tra l'altro ci sarà un ospite di eccezione che è l'astronauta Walter Villadei, che è appena tornato da una missione nello spazio e racconterà come gli studi sulla longevità hanno una fonte molto importante di informazioni scientifiche e biologiche dai viaggi nello spazio. E poi l'ultimo giorno sarà invece sul benessere, la bellezza, l'etica, la filosofia all'Università Statale. Quindi questo è il nostro programma, così vi ricordo che andiamo avanti. E adesso vorrei dare la parola al primi speaker. C'è un bellissimo tête-à-tête tra due grandi scienziati. Li lascio presentare a Nir Barzilai, che ormai voi conoscete. Nir è il direttore dell'Istituto per la ricerca sull'aging all'Albert Einstein College of Medicine di New York ed è l'anima di questo convegno, perché la parte scientifica l'abbiamo creata insieme quando era ancora un piccolo germe, un piccolo seme di idea, e ringrazio moltissimo Nir, perché se abbiamo tutti questi grandi scienziati è soprattutto grazie a lui.
I know you're sincere in what you're saying, but I didn't understand a word.
Ah, you don't have it. I'm saying that you are the soul
when
of
you said bellissimo, it was about you, I know, but the rest I didn't hear.
Okay, but I hope the audience has the translation, and Nir for you, because everybody else understood. I said that you are the soul, the scientific soul of this meeting, and I thank you very much.
Terrific. You're putting lots of pressure on me. Terrific. And welcome to the second day. And I think the first day was quite amazing, and we'll do a little spiral with some new data, some going over older concept, and I think you're here for a treat. So, you know, when people are asking me sometimes, you know, like in the street, so what is aging? So instead of telling them what we're trying to tell you for 5 days now, I say, you know, there's this old couple who has a house in the Middle West in the United States. And the wife turns to the husband and says, honey, why don't we go upstairs and make love? And the husband said, sweetie, I cannot do both. This is aging. Why am I telling you this in Milano? I walked with my wife today and we realized that people in Milano live in apartments. They don't have to walk upstairs. Quality of life here is much better. So it's really great to be here. Great symposium so far. And what we're trying to do in the next less than an hour now is, you know, yesterday you saw that people have data and they skipped it a little bit, because almost everybody that showed you data, this data came from much bigger set of data. We have tests that are capturing hundreds, thousands, 10,000, sometimes 100,000 of data points. And we have to take this data and use computers and use statistics and use mathematics and use physics in order to understand what's going on. So there's no perfect people than the two you'll hear today to introduce you this thing of from computer science to physics and how it can help in longevity. The first, and then we'll sit for a tête-à-tête, and you'll ask question, I'll ask question, but the idea is to give you a test of what we want to do. I have to say another thing, that you know, it's complex, it's different languages, and one of our challenges is to take people from computers, from AI, from physics, and try to speak the same language. And I remember when I heard this lecture about data that was obtained from 3 million people that had electronic medical records over 20 years. Okay, huge amount of data. And they said, we found that obesity is a risk for diabetes. Well, this is not a question, you know, we know that for 60, 70 years. We don't know, we don't need this data. We need everybody to know what questions we ask in order to get the answers that we want to get. So we'll start with the computational science, and I'm introducing you to Naomi. Naomi, come here. Naomi Habib is the Goren Kazam Senior Lecturer in Neuroscience in the Edmond and Lily Safra Center of Brain Science in the Hebrew University of Jerusalem, and that's her title of their talk.
Yeah. Hi, thank you. So it's great to be here today. And let's see. Okay. So today I'll actually introduce not only the motivation why we need these big computational models driven by technology. So my talk will be on the verge between technology and AI, and I'll not dive into details, but rather give you this overview of why I think we have, today, we are in the perfect place to make change, because there's like accumulation of different technological breakthroughs that are actually enabling us to make much more rapid and more dramatic discoveries. So overall, as we age, as you all know, we gain wisdom and memories and knowledge, but we are also, unfortunately, accumulating a lot of damages in our brain, which are the major risk factors for so many different devastating diseases like Alzheimer's disease. And the question we're asking ourselves in my lab, and we're asking ourselves during this entire conference, is how can we maintain a healthy brain as we age? So we'll keep this question in mind and kind of dive deeper into a more kind of general question, and how do we even start to address such a question? So this is kind of how I think of my work as a scientist. So I think about it as like a detective work, right? So we need to gather clues along the way on what is the type of problem we're trying to solve, or disease, to try and identify the triggers of disease, and then go deeper into the mechanism of actual cells and molecules and how they're changing in our brain, so we can design the perfect cure to address that. So as I'll go through my talk, I'll show you how actually technology enables us to advance throughout this kind of schematics. And the four kind of pillars of technology, so you can argue with me, there are so many other technologies being developed, but this is kind of my, where I coming from, my passion, are these four pillars of technological breakthroughs that we've seen over the past decade. So first of all, and I'll go a little bit into more details along these different technologies and show you examples from my own lab and how we use such technologies to understand things about aging and Alzheimer's disease. So first of all, we've gone through a huge, enormous advancement in genomics, in the field of genomics. We're in the genomics era today, and we're generating large amounts of data sets. So these large amounts of data sets would have meant nothing without a kind of a parallel advancement in my field of machine learning, or as referred to as AI, in order to interpret these huge amounts of data set we're generating. And alongside of these advancements, there are two other advancements that are more from the engineering world, enabling us to actually manipulate cells. One is genome engineering, changing the genome, and other one is more cell engineering, modifying cells. And so I'm going to give you kind of a really brief 2 slides, make sure we're all on the same page when we're talking about genomics, so you can follow my talk. So first of all, as you all know, the human genome, or our DNA, is a sequence of 3 billion nucleotides that is stored in our body. And basically, the way I think about it is the human genome is the code of life, right? And then it stores all the information, how to take a sperm and an egg and make an entire organism from it. It also gives us information for each cell in our body, how to function. And unfortunately, also is associated with programming of how to handle different stressors in our environment. And this DNA is stored in every single cell in our body. And as we know, our body contains many different cells, are incredibly different from each other. So how do we translate this huge code into so many different cells? This is because of this basic dogma of biology, where every cell in our body chooses parts of the code to execute. And the code that they will execute will determine the cells that we are forming. So how do we execute a code for a cell? We generate RNA and proteins that are executing the code. So this means that if we want to understand which code is executed, we can measure RNA or proteins. So this is like the intro for the basic biology. Now going back to the advancement. So as I said, the first advancement that we're excited about, that already over a decade ago is the advancement in the field of genomics, literally meaning that we can take a DNA molecule and measure its sequence in high throughput. So if over a decade ago this would have cost for a single genome of a single individual would, actually the first human genome that was sequenced took over 10 years to complete the Human Genome Project, which provided us the full blueprint of the human code. And it would cost over $100 million. This is very unfeasible if you want to take it to the next scale. Today, we're in a stage where I can sequence in my own lab a genome within a day, and it will cost less than $1,000. So this means we can now generate this huge power of genomics to really understand our diversity across individuals and diversity of disease. So let's see how this translates to how can we actually hack our brain or other parts of our body as well. Actually, what apply here is we have our DNA molecules in our body in a little different for each one of us, and we can now have this very powerful tool so we can efficiently measure DNA sequences, and we're generating gigabytes and gigabytes of data. So if you give me a code like ACC, GGG, TTA, right, I can't really interpret anything out of it. So we need to reinforce this with powerful machine learning tools that would help us decode the genomic code and understand it. Okay, so these two advancements in technologies that actually occurred in parallel together come as a very, very strong power. So let's get back to my scheme as a scientist, and how can we actually use these tools of genomics to understand triggers and mechanisms of disease. So starting with triggers, diseases can be triggered by mutations in our DNA. So specific mutations would translate to a different protein that is dysfunctional. And these type of mutations could be triggers of disease. Here you can see an example of the amyloid precursor protein, APP, where a mutation in this gene would cause it to abnormally aggregate, which is happening in our aging brain and in Alzheimer's disease. And a mutation in the APP gene is one of the major causes for early onset Alzheimer's, which is an inherited version of Alzheimer's disease. So this is an example how mutations can lead to could be triggers of disease, and we have so many examples of such mutations. And how can we actually use the power of genomics? We can use that by doing large-scale genome-wide association studies. So once we can sequence many individuals efficiently, we started with cohorts of hundreds of individuals, and today we're looking at cohorts of hundreds and thousands of individuals. And we're basically taking individuals that we know are healthy versus individuals we know are diseased, sequencing their genome in a genome-wide manner, and then using statistics to try and find these specific genetic loci, the specific mutations that can lead to disease. So this is how we can harness genomics to find triggers of disease, but not every disease is genetic, right? We have a lot of diseases, like especially when we're thinking about aging, there's so many factors that are non-genetic here related to environment and lifestyle. So, and also once we find a mutation, it's not enough. We need to go deeper into the cells and the molecules in order to understand the mechanisms of disease. So can we harness these two tools of genomics and AI to understand disease mechanisms? So the answer is yes. We're basically going back to this slide I showed you that every cell is executing the genomic code differently. And the idea is to find how, upon disease, the cells are executing incorrectly this genomic code. Okay, so we're basically going to measure RNA or proteins. And the power of genomics actually enables us, similarly to DNA sequences, we can measure RNA very efficiently. And it's even easier because we have less sequences. So what we're doing is this very simple experiment where we're taking healthy brains and diseased brains, and we're trying to find these molecules of RNAs or proteins that are different between the diseased brain, so we can actually understand the molecule we need to reverse in order to treat disease. So of course, this is a highly simplified view of what we do as scientists for the sake of this argument. So this is the type of work we're doing in the lab. We're taking cohorts of brains of healthy and diseased individuals, measuring their RNA molecules in high throughput, and then using the statistical computational modeling to try and find what are the genes and proteins that might be involved in driving disease. So as I started actually going deeper and deeper into my studies, even during my postdoctorate studies, I realized we have a huge, huge problem when we are trying to translate such type of information using computational models to having actual models of disease. And the problem comes because if we actually zoom into a tissue, a complex tissue like the brain, we see that we don't have a single cell type in the brain, but rather a combination of multiple cell types all working together, communicating with each other, and affecting one another. For example, we have our neurons, our nerve cells that are driving memories and function, but we also have so many supporting cells surrounding them that have a major effect, especially on disease. So it could be our vascular cells, but it could be supporting cells like microglia and astrocytes that are critical for the health of neuronal cells. So what we did is we thought of methods to develop methods that would enable us to harness this huge power of genomics, but still using the right resolution. So we don't want to measure a mishmash of all of the tissue together and all the cells, but rather we want to be able to measure every cell separately and really then use our computational models to separate and understand how does each cell react during disease and change to build much more accurate models. So these are the type of technologies that my colleagues and I have been developing over the past few years, which enables us to profile the brain with single-cell resolution. So this was all very general. How can we actually take such a technology and get some insights about it, about aging and disease? So what we've been focusing in my lab has been on this slow progress of aging and disease in our brain. And here you can see kind of some image of what we've known about Alzheimer's disease as we started our studies. We know that there is a slow progression of the disease. There are accumulation of aggregates of proteins like amyloid beta plaques and neurofilament tangles that are occurring throughout disease. And there is damage to nerve cells in the brain that eventually leads to these terrible, devastating clinical symptoms of dementia, and which is the consequences of Alzheimer's disease. And another very important insight is that actually the disease is incredibly long. And the clinical phase, where we diagnose individuals with dementia, is only the last lag of this course of disease. And in fact, we have over 20 years in our brains where the disease is active, but we don't see any signs of dementia in individuals. So this might be a little scary, because if somebody has dementia at age 75, he probably had the disease already at 20 years earlier, right? So at first it might seem scary, but as a scientist, this was super exciting, and I'll explain why. It's because, first of all, it means that we were looking in the wrong place. So when people have been studying disease for very long and we haven't found a cure, it's always exciting to see that we can actually look at the disease from a different perspective. It gives us a lot of hope for change. So if we kept looking and profiling this last stage of the disease, the clinical stage, we now realize we need to diagnose much, much earlier and understand not the last stage of the disease, but rather the early stages of the disease. So this goes so nicely with what we've been hearing about going to prevention therapy and treating ourselves when we're healthy and not when we're sick in order to prevent the progression to the clinical dementia. Another very nice thing is we actually have 20 years of opportunity here to stop the progression of disease. It's not a single year or half a year that you miss it and you missed the ship has sailed. Okay? Another very important question that we've been asking is, so how is this pathological course different from healthy aging? At which point do individuals with Alzheimer's diverge from individuals from healthy aging? Because this would give us the point where we need to treat individuals. Okay? So to address these questions in my lab, we've been gathering a lot of data from brains, both from mouse models and human brains. So this is actually a big project we've just completed, where we took almost 500 human post-mortem brains from brain donors, and we used our single-cell technology to profile individual cells, and we profiled over 1.7 million cells from these individuals. And we then use our machine learning algorithms to build maps of cells of the brain. So here you can see an example of our map that includes the 1.7 million tiny dots, which each one of them is a single cell. It looks like continents, right? But actually, these are the different cell types that are separating on our 2D representation of the cellular space of the brain. And then we take these maps and try to understand, so which of these cells are related to Alzheimer's, which are related to healthy aging, and how can we separate between them? So I'll actually won't show you data. I will show you 3 slides kind of summarizing our major conclusions from this very, very long work that took us actually over 3 years to complete, because that's the time science takes. So what we found is, first of all, when we looked and we asked which of these, all of these cell types we find in the brain, which one of them is the cell that changes and drives Alzheimer's disease? And what we discovered is that it's not a single cell that is the trigger, but rather what we call cellular environments. Because the cells are so connected and interacting with each other, it seems like they are coordinately changing throughout the course of disease, from a very healthy brain that can help restore itself to a devastating cellular environment that on its own would be highly toxic for neurons and synapses and cause neuronal damage. So this was our first conclusion. The second conclusion is we wanted to ask ourselves, can we actually differentiate between healthy aging and Alzheimer's? And I'll just explain why is this so complicated. Because if we have an individual post-mortemly that we find was cognitively healthy, but had a lot of pathologies in his brain, is he on his way to getting Alzheimer's? We just ended his path 2 years earlier, 5 years earlier? Is it 10 years earlier? Or maybe he's the resilient person who would never get Alzheimer's. He's the super ager, right? So it's very hard for us to distinguish these. But what we realized that what we're doing, building these cellular maps, gives us an enormous power in our hand to ask this question from a different perspective. We're not going to try and ask who is sick and who is healthy, but rather build these paths of cellular change. And this is roughly how it looks. We're aligning cells along these paths of cellular change and trying to find, can we find different trajectories in which our brain is changing? And then go back to the clinical data and ask, is one of this disease and what is the other one? And what we found is that from a very early stage of disease, there's a huge split in the cells in our brain. One path is leading to Alzheimer's disease, like a textbook Alzheimer's disease with amyloids and tangles and the dementia occurring at the end, while the other is a much slower decline of aging, where we see a beginning of accumulation of amyloid beta plaques, and then we do not converge to neurofilament tangles. So we think that what we discovered here is that we can now identify these paths of cellular change, and we know the point where this 2 paths split. So we can now ask, what are these major changes that are driving one person towards the path towards Alzheimer's, and the other one leads to the path for more healthier aging, right? So this is, of course, every research that we do answers a lot of questions and then opens a million other new questions. So we're now trying to understand what might be the major decision point here that is driving us for disease and Alzheimer's, can we use this to do early detection and identify individuals just before the split or at the beginning of the split to prevent them from progressing along the disease, and many other interesting questions. But generally, this is kind of aligns with our vision, which is understanding these early stages of Alzheimer's to go in the line of a more personalized prevention therapy. So I'll skip this because I've took a little longer than I expected. We're also looking how the brain and our body interactions are driving disease. Specifically, our immune system and our metabolism are driving disease. So using similar approaches with single-cell genomics, we can also try and identify not only which diet or what aspects of our immune system are good or bad, we can try and identify the mechanism that mediate these effects. I'm going to skip this for a second, and I'll just talk for 1 minute about options of cure, right? So one option we keep talking about, which we all already have, are these healthy lifestyle that are promoting healthy brain aging, and research like many of us do here, including my lab, really show and explain why would changes in our diet or our sleep will advance healthy aging. But we also have more kind of technological things we can apply. So first of all, since we know how to read the code and we know how to decode it with our models, we can now design specifically engineering tools to correct the brain. So we could use this by either doing cellular engineering. So cellular engineering means we're using small molecules that could induce specific changes in our brain. And there are many such great examples. I think the most exciting ones are the ones where we can actually take cells from our blood or our skin and engineer them into being, for example, healthy dopaminergic neurons that we can transplant into brains of Parkinson's disease patients. But this is like a growing field, and I hope to hear much more about that as we move along with our technology. A completely different approach would say, let's not engineer the cells, but rather go back to our genome and try to correct the actual mutations that we have. So there has been also in the last decade, we've seen a huge transformation in the field in our ability to transform and edit the genome, meaning taking out a mutation and putting in the correct sequence. Right? So this is an extremely exciting area. It's just emerging clinically. We've recently seen a Nobel Prize given to the development of the CRISPR-Cas9 technology. So I was fortunate to work with Feng Zhang while we were developing the CRISPR technology on our own in Feng Zhang's lab at MIT. And the idea of the CRISPR technology is that we're harnessing the bacteria immune system we're engineering it so we can correct the human genome, right? So it's a very exciting kind of way of doing it, and that why CRISPR became so amazing and why this tool was deserving of a Nobel Prize is because it's very easy to engineer, it's very efficient, and it's very accurate. So we're starting to see some CRISPR-based genome engineering in the clinic. The most advanced one is the one correcting sickle cell disease, which correcting blood cells, that correcting the genome and returning them to the patients for a full recovery. But I'm sure we'll see more and more advancement in that field over the next years. So overall, these are my 4 pillars of technology: genomics, AI, cell engineering, and genome engineering, that I think together gives us so much promise and hope to advance science and advance cure. So I'll leave you with my roadmap of hacking the brain, and I'll just say thank you to my lab, who's doing so much and so much fun to work with, many collaborators, and science is a real team effort, and of course the funding sources and the brain donors that we couldn't have done anything without them. Thank you.
Terrific, and it couldn't have been a better example with, you did the buzzwords of, you know, the major leading research and did it beautifully. Thanks so much. So I want to introduce Uri Alon, who's a professor really of system biology, but he's also leading the Sami Segol Aging Center at the Weizmann Institute in Israel. And he's, I think you were trained as a mathematician, right? A physicist. So looking forward to your lecture. That is, read the title.
Mike is there? Mike needs some help? Mike is good, okay. So hi everyone. I always thought as a physicist that aging is very messy, you know, like body falling apart and complicated, and it's not somewhere where physicists, we love simplicity, we're finding simplicity inside complexity. But I discovered it's one of the most amazing and fascinating puzzles I want to tell you about. And I think it's very elegant to think that in aging, unfortunately, we have exponentially rising risk of diseases like cancer. I mean, when you're 70, your risk of cancer is 4,000 times more than when you're 20, for example. So, or diabetes, heart disease, like we just heard about Alzheimer's. And we try to treat these diseases one by one when we get them. But the vision that I find very elegant, and I think many people here, is if we slow down aging itself, we can treat all those diseases together. And that elegance is a driving force. Now, it's complicated because decades of research show that a lot of sources of damage accumulate over age. And this is a schematic that shows that you can have damage in your DNA, and you have damage in your proteins, and in your mitochondria, and cellular senescence that you heard about yesterday, and inflammation, and stem cells aren't regenerated. So all these different kinds of damage, and that's very scary, because to slow down aging, do we have to address all of them one by one, right? This is in order to slow down aging. And what's worse is that they interact. So the DNA damage can make the mitochondria worse, etc. And what are we going to do? Right, so this is a kind of very complex thing. And as a physicist, I guess I was trained to hope that there's simplicity. That is to say, out of all these kinds of damage, there's ones that are more important or they cause the rest, like a red thread. If you can pull on it, you can affect the rest. That's my training. And it makes me see the world in a specific way. Maybe it's an illusion, but... so just for example, I'll tell you why we think that one of these sources of damage stands as a very interesting place between things that happen inside the cell and on the DNA and proteins, and things that happen in the whole body, like inflammation that can drive disease. These damaged cells, zombie cells called senescent cells, are caused by all these kind of damage and affect the body in a systemic way. And that's one kind of way of looking at these sources of damage. Now, as a physicist, the other thing we do is we try to look at a mathematical language of aging. So age for us is t, right? And we just want to slow down the velocity, the t, the way things work. And so using experiments that we did on different organisms, we found out that facts about aging kind of constrain you into one equation. I want to just tell you a metaphor that I'm sure all of you are going to understand about how this works. So you have this damage, we'll think about it like garbage, okay? And it's made by these polluting factories whose number increases with age. And there are garbage trucks. These are removal mechanisms that get overloaded when there's a lot of garbage. And there's stochastic noise or random things that happen, like stress, sleep, infections, that happen during our life, which make genetically identical twins raised in the same conditions still die at different times. So some randomness has to exist in aging. And the way this mathematics work is like this. You have the factories, they're making garbage, and the trucks are removing them, and everything is fine, right? There's some noise, adding and removing damage. But we're not designed to be old. And over time, there's more and more of these factories. I'll tell you what we think they are. And that means you get more and more garbage. But this is a strange country where you fix the number of trucks when you're young, and they don't grow. You don't have more trucks when you have more garbage. We're not designed to be old. And therefore, the trucks are overloaded with garbage, and they can't do other things that they need to do. And then garbage reaches a level that's incompatible with life. That's like our body, right, this metaphor, and we die. Now this, the beautiful thing about mathematics is it's agnostic. You don't need to know what the chemical nature is. It can work for yeast cells and for mice cells, but we can use the model to actually discover what is the chemistry, what is the garbage. We want to know what the garbage is, what the trucks are, so we can address them. I hope I'm being clear. We want to find this out. And using our mathematical model, we have a great tool to do that. We can tell what experiment you do that helps aging actually affects the production of garbage or the removal of garbage, things that we couldn't do before. And our tool is this graph which you do in every experiment on aging. You take, let's say, 100 mice, and you look when they die. So how many survive? And these mice, let's say 50% are dead, let's say 800 days. And then you do something to the mice, caloric restriction or metformin or some good thing, and they live longer. See that? The curve is shifted. Most researchers look at that, how much we increase lifespan. What we're obsessed with is the shape of this curve. That's what our mathematical models tell us. The shape of the curve, when we normalize out the median lifespan, is the curve the same shape or not? That tells us everything for us. It turns out that if what you're doing is affecting the factories, then you increase life, but don't change the shape of the curve. It's like you're stretching time like a rubber band, making time go slower, you can say. You're building less and less factories. But if your intervention is affecting the trucks, you increase life, but now more steep. Everybody dies more at the same time. And the reason is that those trucks make the garbage go away so quickly that noise doesn't have time to randomize things. Things become more deterministic. And as we'll see, that's very good for compressing the sick span also. So now we have a tool just looking at the longevity curves to know if we're affecting the production of damage, the factories, or the trucks, the removal. And now we have new insight into finding out what the core driver of damage is. So I'll tell you about mice. Well, I'll start with yeast, which are amazingly, the things that increase lifespan in yeast, also, those same genes also increase lifespan in mice and probably humans. There's a big conservation. And yeast is so easy to work with. They make babies by budding cells one after another. And what you count is after how many babies the mother cell stops. That's a survival curve. So we got data from researchers that analyzed a huge number of mutations, 30,000 mutations and double mutations. And we looked for mutations that make the curve steeper like this because they affect the trucks. We really want to know what the trucks are. Well, if we know what the trucks are, we can know what the garbage is. See? And it turns out that there are lots of genes that change lifespan, but only very few affect the trucks. They make things steeper. And that was very exciting for us, especially when we saw that those genes tell a biological story that's very clear about basically what are the factories, what are the garbage, what are the trucks, to find the needle in the haystack out of many things that change lifespan into what's a particular driver of aging. And I just tell you what they are. The factories are errors in making proteins that generate prion seeds. These are proteins that are very, very sticky, and they change other proteins to stick to them. The garbage are these gluey aggregates of these proteins that gum up the cell. And interestingly, they go into the cell's energy factories called mitochondria. And that's where the trucks are. The trucks that we found are all mitochondrial garbage removing things, not all the other kind of things that remove garbage in the cell. It's inside the mitochondria. That was new and interesting. And then when those trucks get overloaded, our energy factories get broken and they release superoxides and kill us, basically. So this is a very specific pathway that connects the hallmarks in a specific way. And interestingly, these yeasts have two different ways of dying. It turns out that sometimes they die with these elongated buds with mitochondria damage, but they can also die in a different way with round buds that have DNA damage. So it turns out that, in fact, aging is like an onion. If you remove the dominant source of garbage, you unveil the next layer in the onion, which is the next source of that garbage. And when you live longer, when you treat that, you get to the next source of damage, right? So it's like an onion. And with these double mutations, we can even dig deeper and see what's the second source of damage, the next layer in the onion. And that is, comes from the world of DNA. So the factories here are the DNA is wrapped around these proteins called histones, and there's a specific kind of modification that accumulates with age that makes normally closed areas of the DNA that aren't supposed to be open become open with age. And when that happens, that DNA is turned into RNA, but it shouldn't, it's not designed to, and RNA sticks to DNA and makes these R loops, which are RNA-DNA hybrids. These are dangerous because they can cause double-strand breaks. How do we know that? Because all the trucks we found are these helicases and stuff that remove the R loops. So we found the trucks, the very few trucks that steepen the curve, and we can know what the garbage is. Am I still clear? Okay. And as I said, those R loops cause double-stranded breaks, kill the cell. So we have two ways of dying in yeast. We actually think also in humans, actually, we're guessing, that this pathway with DNA will be in replicating cells, and the one with the mitochondria will be in non-replicating cells, like in our brain, like what Naomi was just talking about, the mitochondrial damage, protein aggregates. By the way, thank you for bringing us together. Two researchers from the same country. We already talked, we're going to work together. So Viviana, thank you. And by the way, when human beings get mutations in those trucks, there are these very rare and horrible diseases of accelerated aging, where a person at age 30 can look like a person at age 70. And this explains why those particular genes, like Werner or Bloom, are accelerated aging genes. So we're really excited to see that, those particular genes. And so far, aging is very complex, but using these mathematical models, we can tell whether we're changing the production of damage or the removal and zero in on the drivers of aging. Now I just want to talk about mice, which are closer to us, of course. Yeah, great, we're doing fine. The factories in mice, as far as we can tell, are cells that stay in our body for all our life, like stem cells that give the rest, make up the rest of the cells. And then by the same pathways as in yeast, histone accumulation that makes double-stranded R loops and double-stranded breaks, the cells become damaged. That's the garbage, those zombie cells that you heard about yesterday. The trucks are the immune system that removes those cells. When you grow old, these cells are great when you're young because they repair wounds, but when you grow old, you have more and more factories. We're not designed to be old. We don't have much more of those immune cells, the trucks, so they get overloaded. Those trucks are supposed to help fight cancer and infection, by the way, but now they're overloaded with those senescent cells, and once they get, the senescent cells cause inflammation and all kinds of things that cause systemic disease and decline. Yeah. I want to end with how this mathematical model can help us with a very burning question. Suppose we have a way to extend lifespan. What we don't want to do is also extend the time that we're sick. So if we want to extend the time where we're healthy. We don't want to stretch time like a rubber band. And if we double lifespan, double the amount of time we're sick, of course. We want to compress the sick span, in other words. But we just don't know which drugs will do that so far, because there isn't good data about the sick span in a lot of organisms. So we can, if we have 50% of our life which is disabled, we can extend lifespan, but also stretch that out like a rubber band, or stretch it proportionally. We want to compress it. We want to compress the time that you're sick. We want to extend the healthy years. And our model tells us something very striking. If you play with the factories, like caloric restriction, you actually stretch time, as I said, like a rubber band, and make the sick span longer. But if you play with the trucks, you have a rectangular survival curve, you compress the sick span. So you're extending life by extending the healthy years. And when we look at mice, which is the closest data we have to humans, we can find, again, that 10% of, let's say, FDA-approved drugs, etc., not only extend life, but also make it steeper. And among those things are senolytics, so that's good. There's drugs that remove senescent cells. They're like trucks. They're supposed to be steeper. That's a good check for us, 4 different senolytics. But then a lot of drugs that affect glucose spikes and like diabetes drugs, SLG2 inhibitors, acarbose. And why is that? Because one of the things those glucose spikes do is hurt our vasculature, our tiny blood vessels that decline with age. And why should that be trucks? Why should blood vessels be trucks? You can think of them like roads for the immune system to come and remove those senescent cells, right? So that's one way to rationalize why those are trucks. And my favorite thing to do with the model is think of combinations. So you want drugs that, you want to combine actually drugs that affect the factories with drugs that affect trucks, because they affect aging in two different ways and they can synergize. So you want drugs that stretch time like a rubber band, like metformin, would extend lifespan, and add to it a drug, like a diabetes drug, that will make the curve steeper, and then you get the compression effects, and they synergize, they add up. So you get the best of both worlds. And to design combinations like that, using a mathematical model is very exciting as a step for future experiments. Combining a truck and a factory gets you the best of both worlds. Reduce the rate of factory production and increase the trucks. So just to summarize, it's a very nice feeling for me as a physicist to at least have a hope for a direction of simplicity inside something as complicated as aging. And this metaphor of factories, garbage trucks, thresholds, very generative for doing experiments. And we think that in yeast we have good candidates for drivers of aging that generate the rest of the damage, and that it might carry over also to mice. And we have a way to figure out which interventions are likely to not only extend life, but also compress the time of life for your sick, the sick span, which we're very eager to test. So thank you very much.
Terrific. So first of all, you demonstrated, as I predicted, that we're not yet talking the same language. And some things you said probably upset some of the people on the conception. But I loved what you said. And I love this idea of, well, the question for me is, okay, if I'm trying to delay the damage and at the end I'm also bringing the truck, is this really going to work? I mean, maybe bringing the truck will increase the damage, you know? I mean, the biology of that is not totally clear, but your examples were something to work with. But we have few minutes. So now you heard each other. You're going to collaborate with each other. Can you tell me what you learned from him that could be applicable to you? I mean, how he can get your data and tell you something that's helpful?
Yeah, thank you. This is exactly what we did in the 5 minutes before our talks. We asked each other what we're talking about and got super excited. Because I think what we've been doing in the lab, we've been generating these huge data sets and then using statistical modeling and these huge kind of probabilistic modeling to try and understand disease. But I think what Uri showed us is that actually trying to simplify the world with kind of principles of physics, and he can explain that better, could help us prioritize, could help us add, like, identify things between correlations to causation. What are the actual triggers? And definitely explore the diversity in our genome as like a huge experiment in humans. So we don't think about it this way, but in mice, we use transgenics, right? We add the mutation, we remove the mutation, we ask, what did it do? In humans, we can't do that. But we actually have a natural experiment in our world, which is a diversity between us. We have each one of us has a different set of genes, or even some are disease-driving, some are just diversifying, and we can try and maybe apply such a model and ask which of these genetic drivers are garbage, using your terminology, are garbages or trucks.
And maybe if you know the garbage in the truck, you can have an intervention that will be timely for the garbage in the truck, right? So how do you look at her data?
Yeah, so it's like a dream listening Naomi to your talk, because it looks like you have data that will have not only the disease-free survival curves for onset of Alzheimer, but you also have the genome and also the gene expression of all the cells there. So this approach, I think, could help us find, hopefully, inside the world of Alzheimer, what is the factories, the garbage, and the trucks. And it could turn out that that garbage is something like neuroinflammation that affects a lot of cells, and the trucks are something unexpected for us that we didn't know, or that things have two functions, both good and bad, you can detect from that. And so once you have a theory, you are hungry for data and for experts that can help you figure it out. And I think we found each other. It's a very nice moment.
So I want to, terrific. I want to get questions from the audience, but I'm sorry, Ana Maria Cuervo, I'm sorry I'm picking of you, but I kind of heard your lecture, right? It was all about garbage and truck and stuff like that. And I'll get you to it soon, but think of something that did you find right or wrong based on your data or applicable. Anybody here with questions? Here, Michael.
Yeah, so thank you both. Great lecture. This is for Naomi. So Naomi, you showed kind of a principal component diagram and with different populations of cells. The neuron cells, it was weird, right? There are a whole bunch of different subpopulations. Maybe not surprising, right? But do you have any insight? Like, are they in different parts of the brain? What are they doing functionally? I'm just curious what all the different nodes there were.
Right. Thank you for this question.
Just
I'll
maybe
explain the
it
question.
better. Yeah.
Okay. So in our cellular maps, we find that all the supporting cells surrounding neurons, like glia cells, are dramatically changing in disease in a coordinated manner. And they're diverging between this more healthier aging and Alzheimer's in the way they respond in their functions. And I was asked, what are the neurons doing? So they're so critical. We know they're dying. We know they're getting damaged, especially in neurodegenerative diseases and in Alzheimer's specifically. How does that go do we have some insights on when is this happening? So I'll say that in our initial work, we haven't found dramatic changes in neuronal cells, and we were very, very surprised. And we actually, it's a nice example of how our computational algorithms change our effects. So what we see is that overall, we couldn't see a dramatic effect initially, and we think it's because the supporting cells have a much more dramatic and rapid change. But when transitioning to, I'll say something more computational, and transitioning from a more kind of discrete modeling way to a continuous way of modeling changes in the brain, we actually suddenly kind of a very strong pattern appeared where we see that coordinately across all neuronal types, regardless of which they are, this is true only for the cortex, because this is where we're getting our data, we see that neurons are changing from the earlier stage. And now we're thinking it might be part of this choice between one path to the other might be rooted in these initial changes in neurons, which were probably reflecting severe damage that are occurring. And this is taking you back to my talk. Probably 20 years before we see dementia, these neurons are already experiencing some damage and responding to it very dramatically.
So, Naomi, but I think, if I'm not mistaken, you refer to the two-dimensional separation of cells where the neurons were they're separated, but they're kind of like that. And you're saying you cannot distinct biologically between them.
Sorry, I gave a really long answer for a simple answer. I'll give the short version. Basically, what I'm saying is that between these two paths of change, we see when we apply the right mathematical model, we see that neurons are probably the initial decision point between going into the path of aging or Alzheimer's disease versus healthy aging. And even though it's very subtle, it's one of these kind of most informative points driving us towards disease.
Terrific. Ana Maria, you have something? I'm sorry, Anna Maria, I owe you one.
Yeah, a big one. Yeah, no, terrific. I think you want me mostly address Uri, right? What he was presenting, and I think it's really exciting way to see things. As Nir said, maybe our definitions of tracks might be a little different, but I think you will be happy to know that one of the tracks that we are looking at, that is this system of autophagy, that is not present in yeast, and that's something that maybe requires a little more of work. Right. It flattens the core. So your prediction that by removing the garbage you get flatter, that's the maximum lifespan is changing more. I mean, the mean that the maximum. So I think that's kind of an example of the type of predictions that you did. And just like one question, because you put it very nicely about it is central for the cleaning of the mitochondria, or at least inside the mitochondria. That's where you brought it, to the factory. So I was wondering if in part that might be a little bias, because the kind of damage that you were looking is mostly mitochondrial origin, but if you have any other kind of damage, whether the cleaning system or the track might be a different one.
So if I understand your question, you're saying that protein damage in the mitochondria is one kind of damage repair, but there's also other ways to repair protein aggregates, like autophagy of different types. Mm-hmm. And what is the role of that? Did I understand your question?
Yeah, the idea is that depending, I mean, are you categorizing your damage as overall cellular damage, or just because you were focusing in particular damage generated by a mitochondria that is not functioning, that's why your outcome was mostly tracks?
Why did we see the mitochondria damage a protein response? So this is a genetics means you do a lot of mutations by random, and you find mutations that do something. For us, it was steepening or shallowing. And it takes you in unexpected directions. You don't know where genetics will take you. And if you get a lot of hits in the same pathway, you say, oh, yeah, I discovered something. So it turns out that in yeast, in budding yeast, the things that make steeper are all the proteins in what's called magic, or mitochondrial removal of aggregates. We got them one after the other. And that was unexpected. And that's the basis for this model, that there are many different trucks, but maybe the first one that gets overloaded, or the first one that young yeast decided to make, let's say 100 trucks, but the others, maybe there's 1,000 trucks for some reason that help the young, that's the fragility that gets overloaded when it's old. So it could be, of course, if you mess all the other trucks, you get accelerated, you get bad things happen. But it looks like the rate-limiting step, according to the genetic screen. Of course, it could be that it's confounded by something.
So in that sense, can you input also changes in different aspects? For example, if you damage the other tracks, I mean, because all these things are always interacting, right? So, and in yeast might be a way, in different organs, different. So it might be a great, great system. I mean, I love your model, because then you can input changes in different points and then see what is the outcome, right?
I think every organism might have its own garbage trucks. Right. And in every situation, calorically restricted have different ones. It's just a way to find them out.
And I think the one thing we kind of, the geroscience is going, we learn a lot from the model, but really, unless you do it in mammalians, you don't have the same trucks, the same damage and stuff like that. So the concept is really great, but whether that's the thing is going more. Thank you, Anna Maria. Thank you very much. Any other questions? Yes, sorry.
I love your model, because the first try that we have to put together the hallmarks of aging, and you pick up the senescent cell as the first layer of the onion, and God knows if you are correct or not. But if you go to real life, today we have a regular patient that is made up of diabetes, hypertension, prostate cancer, dementia, and
poor person.
Something else also, incontinence. How do you put your model in terms of this regular patient, where the senescent cell doesn't look like the first actor of everything.
Got it. So the question is...
The patient is garbage and you have to remove it to the hospital?
Yeah, no, my dear. No.
I think...
Because the garbage is one garbage in diabetes and a total different garbage in Alzheimer's.
Good. So here's the way we think about it. Of course, just having a model doesn't mean it's true. We imagine that you have a load of damaged cells that causes systemic things, inflammation and reduction of stem cells. Okay. Now, whether you're going to get diabetes, cancer, etc., is determined a lot by genetics and your lifestyle. Inflammation and stem cell exhaustion push on all the physiological systems of the body. But according to your genetics and your lifestyle, some of them go into the disease state. Right? When you look at it like that, this model explains why you have exponentially rising incidence of all those diseases you mentioned and falls at the end, because at the end, everybody who could have gotten sick has already gotten sick. So you explained this. And so the way we think about it, of course, doesn't mean it's true, is that you have these one or two primary layers of the onion affecting physiology, and what's going to break depends on your genetics, your lifestyle. And if you're going to get sick with this prostate cancer, you're going to get it. Of course, there's stochasticity, how much garbage there is in each person, so you get it at different times. And for us, this issue of diseases hangs together. Did I answer your question?
You'll go later because we're over time. I'm so excited that there was such a conversation here and so much provocation. Guys, thanks our speakers, and we're moving on.
Is this working? Yeah. Okay. So I think it was very exciting for me, and I think it is for you too, to see science growing in front of you. These are the most advanced research, some of it not yet published, that you're seeing in front of you by these great speakers. And I think it's very exciting to think where science is evolving with artificial intelligence, with new studies, and with ideas that break our normal concept of, what sicknesses are. These, both Naomi and Uri, are looking from a different perspective. And different perspective is what brings to great discoveries. Today is a day of great science, but I thought we need a little intermission to allow you to think of something more curious and rest your mind until we get to the end. And so we thought to make a panel about the people who are behind investments in longevity, and what is going on in this world that is made of scientists, but it's also made of lots of people who spend their lives trying to rejuvenate or to, not age. And so we have a very special panel. We have a very special chair, a young girl, young lady. Her name is Morgane Kendregan. She graduated from London School of Economics with a Master of International Policy, focusing her research on social security system and political economy. Her expertise in working with startups and innovators to bring their visionary ideas to life and help business shape their processes to generate the greatest impact. Thank you, Morgane. Morgane is one of our young chairs. Today you'll have another one. I thought it would be very interesting to have very young people interact with scientists and with protagonists of this world, because, as I say, longevity is their topic. I am too old. Thank you, Morgane.
Thank you.
Oh, you have that.
Oh, I have this.
For the speakers.
Okay. I have this one on my head, so it's fine. Hi, everyone. Thank you, Viviana, so much for the introduction and for inviting me here. It's really cool to be here. In my master's, I remember being in my first class for Social Security. We were doing welfare regimes. And I remember the first day of class, and everyone was, yelling out questions about, you know, like, are we going to have a pension? What's going to happen when we get older? The demographics are, you know, shifting towards an aging economy. What are we going to do? Everyone's freaking out. I'm freaking out. I don't know if I'm going to have a pension. I think these questions for young people are really pressing, and they're kind of at the forefront of our mind. And so it's really interesting for me. I'm hoping today I'm going to learn some things that I can bring back to those students, maybe calm them down, maybe bring up more questions, I don't know. But yeah, I think what's going to be interesting here is we're going to dive into a really fascinating connection between longevity and the economy, and sort of the intersections between the past, present, future of people in this space. Because yeah, longevity, it's not just about living longer. It touches a lot of aspects of our lives, from health, productivity, financial security, overall well-being, and all of the above. So I'm really excited to do this today. We're gonna have 2 keynote speakers, and then we're gonna have a panel and open up questions. So keep your minds engaged and think about your questions, because it's gonna be a good one. So our first keynote is Michael Ringel. We're really excited to have him. As a managing director and senior partner at the Boston Consulting Group, over 25 years of experience under his belt, he's really deeply involved in the initiatives promoting healthy aging. Serves on the board for many major organizations, like Evolution, AFAR, but also really an interesting interdisciplinary background with law and biology. So I think that's going to be quite an interesting talk. So thank you. Let's welcome Michael Ringel.
Thanks for that very kind introduction, and thank you mostly to Vivi for putting me in the young person group. Wow, that is awesome. That's the best thing that happened to me in a long time. And actually, thank you, Vivi, for organizing this great event, and Joelle, and Nir, and Risa. I mean, it's an awesome combination of science and art, and those together are life, which is what we're here about. That's not what you asked me to talk about. You asked me to talk about business, so I will. But before I start on that, actually, I'm going to start on something that's on my mind, because I flew in yesterday from the US, and I'm flying back tomorrow to the US. And so I thought I would start with just thinking a little bit about the effect of circadian rhythm disruption on lifespan. So the Davidson and colleagues take the mice, and they essentially fly them 6 hours east once a week, or 6 hours west once a week. And they look what happens to their mortality. And Nir asked me, like, how do they get the mice to sit still on the plane for that long? Like, is it peanuts and movies involved? I don't think they actually flew the mice. I think they just changed the daylight rhythm. But the unfortunate result of that, if you're a young mouse, actually there's no change in your mortality. But if you're an old mouse, it's fatal, at least flying east. So it's harder to shorten that rhythm than it is to lengthen it. And if anyone has great advice on how to combat this, please tell me before tomorrow, because I'm flying back. My only tricks are I try to get up early when I fly east and stay up late when I fly west, but it's not perfect. Okay, enough about that. What I'm really going to talk about are these 4 things. So number 1, that life and health are immensely valuable, maybe immeasurably so. Number 2, that the major way to extend them, actually the only way to extend them from where we are now, has to be via geroscience. And I'll explain why that's true. Those 2 together create an enormous opportunity. And number 4, it's not too late. Now is actually the time to get in on the ground floor of those. So I'm going to walk you through each of those points, prove them to you, hopefully get you some new data that allows you to think about this more deeply. So number 1, life and health are valuable. So when you talk to people about what we're working on, longevity research, oftentimes you get this worry that actually it's going to harm the economy. And the reason why people seem to have that instinctive reaction are these 2 things. So on the left is that it's going to cost us in terms of entitlements. Old people are going to draw down more sick care. They're going to draw down more social support. and that's going to hurt the economy. And on the left-hand side, the black line is the percent of the US population over 65. The colors are the entitlement spending, and you can see it goes up in line with it, particularly the green, which is the healthcare spend. That's exacerbated by the chart on the right, which is not only do the elderly draw down more, but they produce less. So they're not working, so they're not contributing to the economy. They're maybe not spending as much as consumers. And the data you see on the right there, each bubble is a US state. Sorry, it's a very US-centric page, but it generalizes. But so each bubble is US state, and the regression there is the percent of the population over 60 and the growth in GDP per capita, and you can see there's a negative correlation. And this is the kind of so-called gray wave that you certainly hear politicians talking about a lot. And you know, we have the sort of the boomers at least first to thank for this, but of course it's you know, it's going to be all of us. It turns out that aging research, longevity research, does not exacerbate the gray wave. It's actually the answer to the gray wave. So a lot of serious economists have looked at this, and they all came to the same conclusion. And you know, I'll start first with Murphy and Topol at Chicago. They wrote a National Bureau of Economic Research paper, which is sort of like a gold standard paper in this field. They looked at the changes to lifespan and health span that happened from 1970 to 2000, and they concluded that just that change, historical change, was worth more than $3 trillion per year in terms of benefit to the economy. Goldman and colleagues, using a model from Rand Corporation, did a projection forward and said, what if we changed lifespan just a tiny, I mean, a minuscule amount? They went from the blue curve to the purple curve. If you can see the difference, you know, you've got good eyesight. And they also phased it in very slowly over a 20-year period. And just that very small change, it was worth more than $7 trillion. And then most recently, Scott and colleagues at London Business School looked at a few different scenarios, but you know, one of them was no change to lifespan, just a 10-year compression of morbidity, a 10-year change in health span. And that was, using actually the model from Murphy and Topol, was worth $367 trillion. So, I mean, that's an enormous number. Like, it's so big, it's meaningless to me anyway. Maybe some of you have a better kind of feeling for what that is. To help contextualize it, that's 3 times the entire GDP of the world. So it's a lot of money. There's a lot of value in unlocking this potential of lifespan and health span. So that's the first point. Second point. So I'm going to start here. So this is a Kaplan-Meier curve for humans. This is the number surviving at a given age. And you can see that curve has actually improved over time, over the last roughly 20th century. So much so that we now have a few extra decades of life. That was mainly done through, not actually through medicine, but through sanitation, infant mortality, addressing infant mortality, and vaccination, which I guess is a kind of medicine. It should be readily apparent when you look at that. There's sort of 2 limits. The first is the curve at this point is pretty square. There's not a lot more squaring you can do to it. The second is historically, the maximum hadn't changed. So, you know, if you stick with this as the way forward, there's not a lot of forward in the way forward. That's true no matter how much you spend. So this is a chart of health-adjusted life expectancy versus healthcare spend per capita. Each dot is a country. You know, you can see there's an asymptote, right? No matter how much you spend, you're hitting a wall. And whenever you see a curve that looks like that, you have to ask yourself, well, is there a natural limit that we're running up against? And the answer is yes. That is a natural limit. So J. Olshansky and colleagues wrote a paper in Science now more than 30 years ago that has absolutely stood the test of time. They looked at that and they said, we are going to max out. The increases in life expectancy that we've seen are going to stop. And they did that by calculating, well, if we wanted to get to an average lifespan of 85, we'd have to cure 55% of degenerative disease. To get to an average of 90, we'd have to cure 70%. To get to 105, we'd have to cure 90%. That's in the context of, address mortality rate by 90%. And that's in the context of, even if we were to cure all major degenerative disease, we would only cut out 75% of the mortality rate. So when you look at that, you say it's going to be pretty hard to increase average life expectancy. And that is indeed what happened since they made this prediction. In fact, in some places like the US, it actually went the other way, largely due to opioids and obesity. And you can see their quote on the right, but that's not their actual quote. What they actually said was, unless. They said, unless we have some new technology that can change the fundamental lifespan and health span of humans, this is what we're going to see. And then they go on to say, but there's reason to believe that we may actually be able to modulate that intrinsic limit. And so the way to think about this, which a number of people have referenced, is, so first of all, the number one risk factor for most disease, most degenerative disease, is age. So this is cardiovascular disease, cancer, neurodegeneration, diabetes. These are really the scourges of mankind, and you can see they all go up with age, and they go up exponentially. Now, we haven't thought of age as an intervention point on this, because you live 1 year, you age 1 year. That's just the way it is. But if in fact it becomes possible that you can decouple biological age from chronological age, then this becomes an enormously powerful intervention point, not just to extend lifespan, but to address the burden of disease that we all face. This is the so-called geroscience hypothesis, that by modulating the rate of aging, you can actually modulate disease and a lot of diseases simultaneously. So is there any reason to believe that that hypothesis is plausible? And it turns out actually there's quite a lot of reasons to believe it. So lifespan, and therefore, based on what I just showed you, the incidence of disease appear to be genetically regulated, evolved traits that are therefore manipulable. And there's multiple lines of evidence on this. So the first is theory. Energy and resources are not infinite. Therefore, every organism has a choice of how it deploys that energy, or whatever limiting resource it is, right? Between growth and reproduction and maintenance and repair. And that optimum, which selection will select for, is set by what's going to maximize fitness, not by what's going to maximize your personal lifespan. And the things that maximize fitness are number of offspring, time to them, and the quality thereof. And this theory makes the prediction that organisms in different niches will therefore end up with a different set point, a different optimal lifespan that has been selected for. And that's exactly what we see in comparative biology. Lots of examples of this. I just picked a couple. So the first colorful diagram, which I know you can't read, but that's the radiation of Pacific rockfish. So this is a very new clade of organism. The common ancestor was only 8 million years ago. There is enormous variance in the maximum lifespan across species within rockfish. So some species live 10 years, others live 200 years. Moreover, those differences exactly match what we would predict from evolutionary theory. Changes in depth and size, which correlate with risk of predation and risk of starvation, exactly predict the differences in lifespan. You not only see this across species, but you can see it within species. So the black and white diagram is eusocial insects, so insects that have a queen. In the common black ant, as an example, a worker ant lives 10 months. A queen can live 30 years. They have the same genes, just which genes are turned on and off. So there's a lot of evidence that lifespan is highly manipulable. And then, of course, lastly, a lot of organisms without a soma don't age at all. There's no change in their mortality or reproductive output with age. That's true for a lot of plants. most organisms that are unicellular that don't have a soma. It's even true of some animals that can revert to a germline state, like hydra and immortal jellyfish. There's a third line of evidence around plasticity based on conditions. So pretty much any kind of insult you give an organism, as long as it's mild, makes it live longer, which seems incredibly counterintuitive until you understand it in light of the theory at the beginning, which is if you're in a famine, it does not make sense to reproduce. So you shift those resources to maintenance and repair, stretch, live a little longer, wait for the next breeding opportunity, and hopefully that next season is a good season. And that's the exact surprising result at the top that was discovered in the 30s that kind of kicked this field off by Clive McKay and colleagues at Cornell. They were working on vitamins and nutrition and got this surprising result that when they fed the animals less, in this case rats, they lived longer. And that's been highly replicated across many labs and many organisms. It's not just lifespan, it's health span. So the diagram on the bottom, those are two rhesus monkeys. They are the same age. The one on the bottom has been mildly caloric restricted. You can see it looks much healthier, and it is, by pretty much any biomarker, much healthier. We see this in humans as well. So the top data are epidemiological data from Phelan and Rose across different populations. A negative correlation between caloric intake and lifespan. So that's lifespan data. On the bottom is health span. This guy is a member of what's called the cronies, the people who practice caloric restriction with optimal nutrition. And you can see he looks healthier in the second picture, and indeed by pretty much any biomarker is healthier. We see a line of evidence with genetic interventions. So Cynthia Kenyon's very famous paper that a single gene changed could make a worm, in this case, live twice as long. And on the bottom is human data. Actually, human variations in genes that are involved in the same pathway as these worms are associated with centenarians, people that live 100 years or more. And lastly, we have evidence from pharmacological interventions. So we know it's had proven that you can give a drug or various other interventions to, in this case, a mouse, and make it live longer. This is the first proof. This is from the National Institute of Aging's ITP, the Interventions Testing Program, showing that rapamycin extends lifespan. Lots of other examples since then. The only thing on this page that's missing is have we run a long-term trial in humans to see an intervention extend their lifespan? Now, it's hard to do that, right? It takes a long time, it costs a lot of money, but based on the evidence you see on this page, I think you conclude with a reasonable degree of certainty that if were we to do that, we'd also see the human data as well. Okay, so I think okay, I'll shorten, I'll go quicker. Okay, so I'll go quick through these slides then. So, okay, number one, life and health are valuable. Number two, there's a there with the science. You put those together, you say there's a business opportunity here. And so let me give a highlights of that. So first of all, you see evidence that folks are getting into this field. I'll kind of gloss over this. And that is accelerating even in the last 2 years. So all the kind of leading indicators, things like media mentions, publications, company formation, investments, not only growing faster than background rate, but now ticking up in the last couple of years. As a result, we now have more than 200 companies with more than 10 billion in investment, with a reasonable sized pipeline in clinic now. This is in trials. And those assets actually cover a huge range of therapeutic areas. For a relatively small pipeline, and this is exactly what you would expect based on the geroscience hypothesis. So if modulating the rate of aging can actually affect a lot of different diseases, you would expect to see a fairly broad spread in the therapeutic areas covered, and that is what you see. And we also see pharma companies getting interested in this space. So multi-billion-dollar investments. This is largely through deal making, not through units themselves, although they've Novartis and Regeneron have some small units. And I think in 5 years, we're going to see actually quite a lot. Okay, and then lastly, quickly on the it's not too late to get in, just to give you a sense. So there's activity ticking up, but we still have a long way to go before this is like a saturated market. So I'll compare it to oncology as an example, which is an area that still needs a lot of work, but it's more mature. And you can think about metrics, right? So there's a chain of activity that has to happen. There has to be funding so that scientists can do the work. Scientists have to come up with findings so that biotechs can work on something. Biotechs have to get assets moved into the clinic that pharma then eventually picks up. And so it's like a snake swallowing, it's going to take some time for things to move down this chain. And so we would expect to see, given a recent uptick in work on aging biology, things to be kind of closer to parity up front and then move further away from parity as you move down the chain, and that's what we see. It's actually still way off in the first instance, so we've got about a 10x difference in public funding, but a 15 to 20x difference in academic output, something like a 40x difference in number of companies, and more than 100x difference in pharma OpEx. So, even oncology, it's not too late to get in, but you're by no means late if you want to form a company in this space. And I'll just close on one more slide, which is a lot of you are here not just for company formation, but for personal interest. And so to kind of make that real, I thought I'd just share a recent study that came out last year. This is the 8 habits that could extend your life 24 years. It showed up in a ton of different publications, so you probably saw it unless you were soundly sleeping. But if you did miss it because you were soundly sleeping, that's okay, because it is one of the 8 habits. And you probably want to know the list, so I'll just go through these quickly. So there's the Holy Trinity: exercise, diet, and sleep. You don't need to exercise that much. Actually, the benefits of exercise accrue asymptotically. So even a little bit, in this case, the threshold was at 15 minutes of run a day. Even a little bit actually has a huge benefit. So if you're not exercising, absolutely start. Plant-based diet, not too much. Good sleep. Then there's the 3 thou shalt nots. Don't drink, don't smoke, don't do drugs, don't have any fun, right? And then there's two mental health related ones: stress and social relationships. And there's a whole bunch of stuff they didn't test here, right? All sorts of drugs and other supplements you can do, some of which are prudent, some of which I'd say not so prudent. To maybe give an example of the latter. Eunuchs achieve centenarian status at least 130 times the background rate. I hope this word translates. This, by the way, this is recapitulated in other species. If you ablate the germline of a worm, it will live longer. Spayed and neuter pets live longer. It matches theory in terms of how you allocate resources, but again, not something I would suggest. So let me stop there, and we'll go on to the panel.
Thank you so much. That was great. Extraordinarily interesting to get a whole overview of investment and finance and where we are headed in the industry, which ties in really, really well with our second keynote speaker, André Bertram. André is a dynamic entrepreneur and philanthropist, currently serving as the chairman and president of Bertram Endeavors, with a focus on healthcare and blockchain. His leadership's been really instrumental in incubating and investing in innovative companies, and is former CEO of Helpware, so giving us also a good idea of startups and all of that. So let's welcome André Bertram. Thanks.
Beautiful. Thank you all for being here. Today you guys have all had the opportunity to learn from some of the world's leading researchers in the biology of aging and the math of how to understand it. Michael Ringel, who I didn't know I would be following, I think he did his bachelor's at Princeton, he got a PhD in biology, and then he studied law at Harvard. And now you guys get the opportunity to hear from a mid-20s college dropout. So you can thank Viviana for that opportunity later. But I'm not here to talk about the science at all. There's many other people in this room who are far more qualified than I am to do that. I'm here to talk about giving money in this space, why I do so, and why a lot of other young, very rich people have started to do so as well. And how we're trying to better build the industrial complex, or the pipeline, to get some of these therapies from ideation through to the clinic. So first, a few disclosures. One, I have controlling interest in a global medical device company. I'll do my best to not sell heart monitors during this presentation. Don't worry about that. Secondarily, I have ownership in about 250 other private companies. I own everything from a gaming company to a large bookstore chain. Again, won't be a problem here. Beautiful. And then an important note is that none of them are explicitly longevity companies. And I find this to be incredibly important to me. My engagement in this field is solely philanthropic. I do not have a company. I have nothing to sell you. I net give away money in this space, which makes me have a little bit of a different perspective than some of the other folks you've heard from so far. So a little bit about me. I'm a 26-year-old entrepreneur and investor. I live between Toronto and Abu Dhabi, which is an emirate in the Middle East. I dropped out of MIT and Waterloo when I was 17 because a billionaire named Peter Thiel paid me to leave university and start building a company. And I had a goal from when I was 16 years old to try to save 1 million lives by quality-adjusted life years before I turned 30. I've got a lot of catching up to do. So how did I start that? When I was 16 years old, I founded a medical device company called Helpware in the cardiac remote patient monitoring space. We built ECG monitoring equipment to predominantly stop cryptogenic strokes, which is a type of stroke with no known cause. And very quickly, that got a ton of media attention. Before I knew it, we had offices all around the world. We were one of the fastest growing companies in Canada. We partnered with Johnson & Johnson and scaled across China. We went into the UAE on invite from the crown of both Dubai and Abu Dhabi. And by my early 20s, I was running a global business and, had done quite well for myself financially. And luckily, along the way, I started investing in some of my friends' businesses. And this graph right here is the homogenized average yield of both technology businesses and crypto from 2015 when I started investing. And that's when I started deploying all my money into tech. So it really was more luck than it was anything else, but I was fortunate that I had done very well at quite a young age. So by kind of my mid-20s, the last few years, I've had a resource that I didn't have when I started my first business, which is access to a lot of my own money that I didn't have to ask anybody else for. But I had a problem. Medical device was too slow. It took me 8 years to get my first device from ideation through to scaled manufacturing globally. And every time we sell a cardiac monitor, on average, we're extending a patient's life by about 11 quality-adjusted life years. So you don't need a PhD in mathematics or statistics to know I would have had to sell a hell of a lot of heart monitors to save 1 million lives. So I had this friend, Laura Deming. She's a lot smarter than I am. She did her PhD at MIT when she was 16, and Peter had also paid her to leave school. And she had raised millions of dollars to start Longevity Fund, which was really the first VC firm in the space. And what she was telling me about was these researchers, like this weird guy, Nir Barzilai, who had started calling themselves geoscientists. They were better trying to understand the actual biology of how humans age so we could better understand how to slow it down. And this was very interesting to me because there were dozens of scientists just like Nir. And Nir had a belief that metformin, a generic drug that costs less than 13 cents a day, could meaningfully extend health span. And I would highlight that this hasn't fully been proven yet in a large-scale clinical trial, but if a drug at 13 cents a day had a chance of moving the needle, there was clearly a more effective way to be spending my time than a med device. So I had this question. Why isn't there a drug to slow the rate of aging? There was more research in this space than oncology had when they were already deploying tens of billions of dollars into the field globally. So I went on a mission to identify and help eliminate the barriers that were facing commercialization in geoscience. So I started a process. Over a period of 14 months, I started interviewing some of the world's top researchers, a lot of which are here today, as well as the top nonprofit organizations, the American Federation for Aging Research, SENS, Methuselah, anyone who is giving away a large sum of money in the field. There weren't that many. As well as some of the donors and entrepreneurs who had already started to build businesses in the space. Sami Segal, who's here today, has been a prolific donor in both brain health as well as the biology of aging. And folks like Jim Mellon in the UK and Christian Angermeyer had both raised hundreds of millions of dollars to build billion-dollar private businesses in the field. And we really determined one core thing, which is that geroscience is missing the pillars of a therapeutic industrial complex. So what do I mean by this? So let's say you want to take a drug to market. What are you going to need? Well, the first thing is you need a bunch of reputable legacy grant makers, right? So let's say you're starting a cardiac therapeutic, right? You know, a grant from the American Heart Association would give consumers and investors trust that this research was genuine and authentic. Next, you need an institution for sense making, some group of credible people to tell you which things are authentic and which things aren't. So, you know, the American College of Cardiology or the Heart Rhythm Society, which is the big association of electrophysiologists, would say, this science is real and good and is to be acted on, this science is not. Next, you would need an industry association. It's really important to get all of the largest companies in a field talking together so that they can address, you know, pipeline issues for hiring talent, so that they can address policy issues that are blocking them. And that feeds right into a lobbying organization. The #1 inflection point in a medical device business is when you've convinced the American Medical Association to give you a new billing code or add you to a formulary so that you can sell across the United States. We weren't even at the phase where the industry had formed together in longevity when I first started giving money away, let alone at the point where we were lobbying governments effectively. And then lastly, you need a large capital base for investment. There's tens of billions of dollars deployed by private venture firms globally into general therapeutic areas. None were explicitly investing in gerotherapeutics. So this gave me a ton of anxiety. I had this overwhelming relief that I was running out of time. If it was going to take 8 years for me to get a heart monitor through the FDA, and they didn't even have an industry association formed yet, I was starting to think that even though I was 26, I might not live to see some of the innovations that Nir was working on in the lab. And a lot of other young folks like myself felt the same way. And from that day onward, I've started donating to the field. But I wasn't alone. Here are some of the other young folks that have been deploying capital. This is James Fickle. He's very, very private. He was a software engineer who used to just build mobile apps for companies that didn't have apps. And in 2015, he sold everything he owned and put it all into a cryptocurrency called Ethereum at about 86 cents. And now he's a multi-billionaire. In bear markets, when the market's down, he donates $25 million a year to aging research, predominantly brain aging. And when the market's up, he donates $100 million a year. He founded the Time Initiative, which is now at the Academy for Health and Lifespan Research, which helps fund undergraduate researchers who are interested in the field in coming over and spending a summer focused on aging. Next, you've got Jed McCaleb. He's the oldest of the group at 49. He founded a cryptocurrency called Ripple and another one called Stellar, and his foundation, Astera, has committed $1 billion to aging research. He donated $70 million to the Buck Institute to help better understand mouse models. You've got Vitalik Buterin, 30 years old, another Thiel fellow who Peter paid to leave school, a little bit of a trend there. He started VitaDAO, which is essentially a decentralized organization where people can donate money online and all get to collectively vote which foundations or nonprofit it supports, exclusively for aging. And he also started Zuzulu. Zuzulu is a partnership with the government of Montenegro. It's a full longevity city that runs for 3 months in Montenegro. And he's working now with the government to help affect policy to make Montenegro a longevity region. And then lastly, you've got Juan Benet. Juan's 36. He founded Protocol Labs, which released Filecoin. He's donated over $100 million to the space and was the anchor investor in Impetus Grants. Impetus Grants is fast grants for longevity. So if you want a grant from them, you write something up, you send it in, and they decide within a week or less. But if you think these billionaires have started to move the needle, wait until you meet the trillionaires of longevity. This is Prince Mohammed bin Salman, the Crown Prince of Saudi Arabia, and it's important to note that he's only 38 years old. This is a very interesting paradigm shift, right? Because the difference between a 38-year-old leader thinking about investing in this technology and somebody like, who might be running the United States, for example, is MBS has the opportunity to actually see some of the drugs that he's helping build go into the clinic to affect his patient population. And this is really important because there's over 40 million Saudi Arabians, and the average age of a Saudi is 30.6. So not only is making a commitment to this field something that MBS is going to be able to see, but it's something that the bulk of its country is going to be able to see, to live longer and to live healthier. That's why he endowed Hevolution. Hevolution's a commitment of $20 billion US or $1 billion a year in perpetuity to fund the field. he's not the only one. Back at my second home in Abu Dhabi, which, I mean, I've got 2 homes that are 14.5 hours by plane away from each other, so I hope Michael Ringel's at least a little bit wrong. But this is Sheikh Sanoun. He's 55, so, you know, a bit on the older side for an Arab leader. But what's important to note is one of his reasons for following actually had nothing to do with the fact that Saudi Arabia had started building a foundation already. There's a line in the Quran that states that if you were to save the life of one, it would be as if you saved all of humanity. And in a lot of ways, the UAE royal family has a deep sense of commitment to trying to affect lives with their wealth. So there's about 10 million people in the UAE, only about 1 million of them are passport holders, but of those million actual Emiratis, the average age is 33.6 years old. And Modera, the $380 billion sovereign wealth fund of the government of the UAE, has now committed $500 million to start a longevity fund under Modera Capital, which will invest in early stage longevity companies globally. So, where are we now? It's been about 2.5, almost 3 years of myself and a lot of others giving away capital to try to build this out. Well, when it comes to a reputable legacy grant maker, we've got the American Federation for Aging Research. It's a 40-year-old foundation that's been around. It started by a bunch of insurance executives wanting to get a better understanding of how humans age, which can really act as that trusted entity for governments and partners. When it comes as an institution for sense-making, the Academy for Health and Lifespan Research was started by Sami Segal and 15 of the world's top aging researchers in 2018 in Paris. And it's now scaled to 60 full members and is really kind of the premier source of longevity research talent globally. Next, in terms of an industry association, Jim Mellon, that good-looking Brit from the earlier side, and James Pryor from Cambrian started the Longevity Biotechnology Association, which is a fast-growing industry association for the early-stage private companies in the field. When it comes to lobbying, A4LI is starting to be the first discrete lobbying entity in the United States explicitly for benefiting geotherapeutics and geotherapeutic companies. And then lastly, in terms of a large capital base, you know, just between Hevolution, Modera, and Milky Way Ventures, you've got a commitment of about $1.8 billion a year in private invested capital. So I'm hopeful that in my generation, we'll be able to have a long and healthy life that's accessible to all. Thank you.
Thank you so much. Thanks. Thank you, that was amazing. Okay, so we're short on time, but we're going to have the panelists come up. Ivan, Camilla, and Sam, if you could come and take a seat. There you go. Yeah, whichever one. You can sit there. Come here. Here, you can sit next to me. Great, thank you. Okay. So we've had amazing introductions from our keynotes, and now we're going to dive a little bit deeper into the people that are testing these, let's say the guinea pigs, and how, yeah, what are they like, what drives them, and their experience. So we have Ivan Manandola, one of the first people in Italy involved in the longevity space over 15 years ago, before it was really a topic on people's minds. Is now the administration delegate for Precision X, a fund dedicated to longevity and precision tech. We have Sam Venice, a writer and now founding editor at Project Glitch, who's been learning about a city, Vitalia. It's sort of a hub of cryptocurrency and experimental technologies. And so I'm really looking forward to hearing more about his experience. And Camilla Baresani, a writer and journalist who's spent time living in, well, seeing the sort of communities in the US that are also living a lifestyle of longevity through fitness, nutrition, and new technologies. Great, so starting with Ivan, I mean, yours is a wonderful story of passion for the space and pioneering, one of the first here. So I'm curious to know a little bit more about your experience, the difference between the space then and now, and the people involved.
Well, hello everyone. Well, my story started back in 2002. I did my MBA in Boston, one of the big Ivy League schools, and I was sent over. The decision was to go back to banking. I'm a finance guy by training, or go to Singapore and meet this random guy who back then was basically a nobody. His name was Craig Venter, who formed Cellular Genomics, who basically gave birth to the Human Genome Project, 2001, 2003. And so I spent 7 months of my life in Singapore. Back then it was the early days of next generation sequencing, genomics. It was the early birth of the omics science, and I was, let's say, bred in a very scientific environment. My dad was the lead doctor of the medical programs in all over Africa. So we spent over 30 years in Africa as diplomats, but my dad was basically opening medical wards. So what happened back then, short story long, I went back, decided to give up my career in banking, and basically started the first generation sequencing company back then in 2003, raised over 50 million. Technology was based in Estonia. Maybe some of the big scientists here know the name, big professor Metspalu in Tartu, Estonia, which is a big molecular biology school. Back then we were basically competing with another company, which was Decode Me, an Iceland company. And back then it was only about genomics. It's funny because I'm not, of course, going to go into science at all. I mean, Viviana asked me to bring my experience and some maybe anecdotes of what was back then, which I call the first republic of anti-aging back then personalized medicine. Now it's more, I like it to refer to it more into precision, you know, precision medicine, precision health, which longevity is a part of it, right? So it's funny because I was then in Silicon Valley, where I spent more than 12 years. We were having lunches with Anne Wojcicki, which is the ex-wife of Sergey Brin, the Google guy, which she started 23andMe, which is one of the biggest, largest nowadays direct-to-consumer genomic company. We were all trying to understand, you know, back then there was no RNA, there was no epigenetics, so we were trying to understand how to sort of understand the biology of cell and the gene expression. And we had a I remember a very funny lunch in Palo Alto. I mean, there's a lot of friends here from Stanford and the big schools. We were with Lars Rasmussen, the two brothers that maybe the name ring a bell for somebody. The two brothers who invented Google Maps, who then was sold to Google. And back then, the very inspiring example we had is that back then genomics, and I loved Professor Habib's presentation because, of course, you know, it's what we have lived since 2003. Is that back then we only had one picture, which was DNA. I mean, if we take our slide back, we didn't have all the protein and RNA parts. So Lars, which was just about to create Waze, which then became Google Maps, and he was like, well, it's like having basically a Google map with the satellite not being able to tell you, you know, when the road becomes, you know, there's work in progress, or the road turns, you know, the size, or the direction, right? So fast forward, then I spent a lot of time investing in myself in medtech. Similar to Andres, I mean, lovely story. And I'll close my intervention. The funny part is that I was drawn back in the longevity space. 2 years ago, we had lunch with David in Boston, David Sinclair. And so I basically gave him my sort of I call it again the first republic of longevity, and it sort of filled me up with this, the new trends of health optimization protocols and technology, consumable device, and that's why I started putting again the joining dots and given my experience in startups, tech, device, we made similar investors like André. We did Misfit. We did Wheatings. I was working with 500 startups. Another funny example, we had so many lunches with Sam Altman, the ChatGPT founder, but he was also the founder of Y Combinator, which is one of the largest accelerators in Silicon Valley. And he also heavily invested in the space right now with Retro Bioscience, which is a similar competitor of Turn. So yeah, so.
Great. Yeah, and I am curious, Camilla, about your experience. Obviously, we were touching on a bit of the past, but you are someone who just experienced what's happening right now in the present with the group. So I'm curious to hear a little bit more about your experience there.
Bene, anzitutto io rivaluto l'uso del vituperato italiano e per prima vi parlerò in italiano. Vi leggo brevemente una cosa. Molti di voi sanno chi è Hokusai, un grande artista, un pittore giapponese, che nel suo testamento spirituale ha scritto questo: dall'età di 6 anni ho sentito il desiderio di dipingere qualsiasi cosa vedessi attorno a me. Dopo i 50 anni avevo già fatto un buon numero di opere, ma non ero affatto contento del mio lavoro. Solo adesso, all'età di 75 anni, ho parzialmente capito la vera forma e il carattere di uccelli, pesci e piante. All'età di 80 anni avrò certamente fatto ulteriori progressi, così che quando avrò 90 anni riuscirò a penetrare nella vera essenza delle cose. A 100 anni raggiungerò un livello di perfezione e all'età di 110 ogni cosa che io creerò, ogni punto e ogni linea che traccerò saranno vive. Io invito tutti quelli che allora mi conosceranno ad accertarsi della verità di queste mie parole. Ora, poverino, lui non ha vissuto fino ai 110, quindi non abbiamo potuto verificare se effettivamente i suoi tratti, i suoi disegni erano diventati vivi a 110, però ha vissuto fino agli 89 anni. Forse era una di quelle zone blu che oggi studiamo, dove si vive a lungo. Quindi questo sogno di longevità è molto vecchio, è secolare, è il sogno non solo degli artisti come Hokusai, ma di ciascuno di noi. Un'ulteriore piccola cosa volevo dire: parte del sogno della longevity declinato al femminile, e ricordiamoci che la scienza, l'università, l'impresa, l'industria sono ancora molto mondi al maschile. Ricordiamoci che la biologia femminile è inclemente, è anche ingiusta se vogliamo, quindi parte di questo sogno della longevity femminile è il social freezing, cioè la crioconservazione degli ovociti, che permetterebbe a noi donne di prolungare la longevità del nostro corpo, ed è una cosa importantissima, se ne parla molto poco, ma sarebbe un modo di riparare a questa ingiustizia biologica che noi donne patiamo e che un mondo ancora molto maschile ha magari trascurato. Quindi diciamo che auspico che tutti gli scienziati che sono qui presenti, gli studiosi e anche l'industria si occupino di aiutare le donne a superare questo gap e questa ingiustizia. Detto questo, arrivo alla tua domanda. Ho avuto modo di interessarmi al problema della longevità 2 volte. Una volta ero in California, sono stata a lungo nel 2016 a San Francisco, dove ho potuto parlare con molti start-up, con molte persone che lavoravano a sviluppare, a potenziare questo sogno. Ma la cosa che più mi divertì fu parlare col cuoco di Peter Thiel, che molti di voi conoscono come uno dei più ricchi imprenditori della Silicon Valley, il fondatore di PayPal, il secondo socio di Facebook, e anche un uomo che spinge i sogni all'impossibile, tipo creare delle isole che siano delle città perfette dove vivono poche persone senza pagare tasse e vivendo all'infinito. Ecco, il cuoco di Peter Thiel mi svelò come viveva Peter Thiel, cosa mangiava, e capii che quest'uomo aveva un'attenzione spasmodica sull'alimentazione, ancor più che sullo sport, sull'alimentazione e sugli integratori. In quel periodo, per esempio, era fissato con il colore dell'alimentazione, quindi si mangiavano solo cose di un certo colore. Dopo aver visto e osservato questo mondo dell'attenzione alla longevity nella Silicon Valley, in California, molto volto anche a risolvere un problema sociale, cioè il costo sociale dell'invecchiamento, del prolungamento della malattia, della longevità malata, mi è capitato invece di vedere dall'altro versante, il versante East Coast, il problema, cioè nella fattispecie a Miami, dove vive uno dei nostri guru, che è stato ospite il giorno 14, Camillo Ricordi. Ecco, lì c'è un versante che io chiamerei gli egoisti, cioè nessuno si interessa a finanziare e a creare un movimento di interesse scientifico e imprenditoriale al tema della longevità, pensando a tutte le persone che muoiono presto per la povertà, perché mangiano troppo, perché mangiano male, perché non hanno tempo di fare esercizio fisico, ma ognuno è concentrato su se stesso a livelli spasmodici, una sorta di malattia quasi psicologica, ho notato, cioè come una nuova malattia, una nuova religione. Noi in Italia, non so, dicevamo, ce la prendevamo con i bigotti, con le persone ossessivamente religiose. Ecco, lì è un'ossessione quasi religiosa, l'ossessione dell'integratore. Ho anche riso vedendo quante persone erano fissate con la stem cell therapy, un costoso trattamento di cui ancora non si sa se i vantaggi che si possono ottenere a breve possano diventare delle malattie mortali alla lunga. Eppure tutti erano concentrati su fare questo ricambio delle cellule del sangue, e per risparmiare alcuni vanno in una clinica a Tijuana, che lì va per la maggiore, perché il trattamento costa la metà. Quindi ho potuto osservare appunto questi due versanti. L'America è ovviamente per noi italiani il paese a cui sempre guardiamo per le rivoluzioni scientifiche, per le rivoluzioni anche sociali, e così questo mondo degli egoisti di Miami l'ho trovato socialmente, cioè per uno scrittore, per un giornalista come io sono, un mondo anche divertente, paradossale, assurdo, come nuova forma di vizio mentale della società dei ricchi.
Interessantissimo. Sam, basically, you don't speak Italian, I'm assuming. Or?
I saw lots of smiling faces and nodding heads,
okay.
but I did not understand the word.
So I mean, Camilla was telling us about her experience in America, in Miami and California, with people that are what she describes as obsessed with the lifestyle of longevity and maintaining it. And she saw it very much as like this self-absorbed obsession. But she found that quite fascinating, that actually it's quite a really interesting place. And I think that perfectly leads on to you because of your experience in Vitalia. So I'd love to hear more about that and just the sort of modern day fountain of youth and what your experience there and what were the people like and what were driving them.
Yeah. So to give some context, I just came back from a city in Honduras on the island of Roatan called Vitalia, which was basically it was a pop-up city in a special economic zone called Prospera. Prospera is also, to sort of build off what André was talking about earlier, is another venture backed by Peter Thiel, in which basically it's a jurisdiction where companies can be formed and be legislated according to whatever rules they choose, including ones of their own making. And so that sort of spawned, it's being used as an impetus to drive people there who are interested in medical innovation and biotech more generally. And it's hard to tell whether that's based on interest or if it's because of the opportunities that are developed because of having sort of a regulatory zone where you can be a little bit more, let's say, innovative with the approach. It's it's very interesting sort of seeing the contrast between that world and this one, because this is very institutional. There's amazing science being done here, but there's a lot of serious money that's involved and funders. There, it's kind of a little bit more ragtag. There is a lot of money that's being invested, and a lot of it, as André kind of identified, is a lot of young billionaires who, many of them are in crypto, which sort of brings a mentality that's a little bit anti-institutional by nature and sort of looking for ways of like going outside of the system. And whereas I think because of the nature of that, you have in this room a lot of people who are making more modest claims about what longevity science can do and thinking about quality of lifespan, thinking about extending, maybe to 120 if we go wild and figure some things out. There are a lot of people there who are talking about doubling the human lifespan, living forever, and there's people that are extremely passionate about it and thinking about it and allowing their imagination to roam free. So there's people, for example, I met someone who is looking to do research on head transplantation, on having to figure how to keep a head alive outside from the brain, which if you're trying to, as we develop artificial limbs, if you're trying to solve this question of how do you end aging, these types of radical approaches are maybe something that you would be interested in. And because of the nature of this economic zone in Honduras, where although, where you can kind of experiment and push the limits a little bit on what's possible, let's say, in the EU or in the US, it's attracting those type of people. And for better and for worse, I'm sure, And I think what's interesting for me is seeing the sort of, because of these two worlds that are sort of colliding and using the same banner of even just the word longevity, it's an interesting sort of mix, because there's this, on the one hand, mixing of ideas and science and passion and media attention, but that comes with its own sort of risks of how do you make sure that if something bad happens, if somebody dies, it could set back the field in a significant way. So it's a kind of interesting sort of dialectic between two parts of something that are focused on the same goal, but approaching it in very different ways.
Great, thank you. Unfortunately, that's all we have time for, but I'm sure you guys will have loads of questions, so please find everyone that spoke afterwards to ask them all your questions. And thank you so much for all of the speakers. Thank you.
Okay, so we heard a lot of how to prolong healthy life, maybe extend the lifespan, about people who are involved, the market, the longevity market, the selfish longevist, and the people who are philanthropists and do it for humanity. And we are now at our last panel, that is the future. And we have a young chair again, Nicholas Kazam. He's a Fulbright recipient. He's a Master of Science and Journalism from Annenberg University in University of South California. And again, a young speaker like Morgane before, because as we also heard from André, it is the young people for whom longevity will work, if it works, and who can hope to expand their life and to live healthier. Sometimes for us, it is a dream. We hope something will come up fast, but you know, research and medicine require a long time. And so it's this generation who hopefully will live healthy and maybe not aging until 120 or more.
Thank you so much for that introduction, Viviana. I really appreciate it. I would just like to say, first of all, thank you to you for being here. Thank you to everyone in the audience. Thank you to everybody listening online. But most of all, thank you to everybody that played any part in making this the most exciting longevity conference in the world. It's truly been an honor to be here. The past few days, I've gone to meet many of you, and so many bright minds in this room, so much exciting work being done. And as a member of the younger generation, who I suppose I'm representing here today, I just want to say that you've inspired me, and you are continuing to inspire young people around the world to be happier and healthier and to live long lives so that they can continue working on exciting things for years to come. So this panel is called The Peter Pan Effect, posing the question, let me just make sure. Yeah, it is. It's called The Peter Pan Effect, posing the question, will we be forever young? As you know, Mr. Pan did not have access to the groundbreaking science that is being discussed here today, yet somehow he was able to attain a remarkable health span. And I'm going to be introducing two very exciting speakers, Nir Barzilai and Vittorio Sebastiano. They're going to potentially answer the question, can we be forever young? Is it a reality or is it a fairy tale? So first, we are introducing Nir. He is the director of the Institute of Aging Research at the Albert Einstein College of Medicine and director of the Paul F. Glenn Center for the Biology of Human Aging Research and the National Institute of Health's Nathan Schock Centers of Excellence in the Basic Biology of Aging. I don't know if you've gotten a chance to meet him, but he's also a really funny guy. Nir, thank you so much and welcome to the stage.
Thank you. And Nico, you know, I want to say we want to kind of stop, maybe reverse aging. Some of us are saying that. But if you look at Viviana and Sami Segol, who are, you know, exceptionally wisdom and brain power and innovation, when we reverse aging, we don't want to reverse that. Okay, we don't want to reverse the wisdom. Maybe Naomi Habib, we should talk about it. Okay, this is always on my mind. Are we? It's like what's the movie that got the Oscar? No, just now. Poor Things. Poor Thing. Okay. Okay. Anyhow. So I'm going to do two provocations in the next 20 minutes, okay? One of them is going to challenge a repeated sentence that you said that we're far away from treating people. It's not true, okay? And I want you to challenge me if you think that I was wrong at the end of this talk. And then there's a second thing. You notice that instead of David Sinclair, we took somebody who's younger, better looking, and although he's in a department of obstetrics and gynecology, you're probably the smartest professor of OBGYN in the world, okay? So I'm so happy that you're coming later. And, you know, we all prepared some slides knowing that somebody else talked about that before, so I'm going to really go fast through some of the slides and just make some points, right? And don't forget that life expectancy through most of evolution was 20 to 30 years old, okay? That was life expectancy. It's really in the last 150 years that we made this progress. And it's during this last 150 years, when we got to the age of 50, that we all of a sudden got disease after disease, treatment after treatment, and we end up with a bad quality of life. And those diseases were not part of evolution. So somebody came to me and said, you know, I have prostate cancer. How come evolution didn't take care of that? Well, people didn't die from prostate cancer before, okay? Aging is very post-evolutionary in many ways, and certainly because life expectancy didn't allow us to really deal with some of the problems we have now. And it'll probably never will. We have to do it ourselves. But what happened in those last 150 years? It's mainly prevention. Okay, it's mainly prevention. It's harnessing the agriculture, it's cleaning the water, it's building sewers, it's vaccination, okay, it's lowering cholesterol, okay, it's mainly prevention thing, and that's how we have to talk about aging. We don't want to deal with diseases anymore, okay? We want to prevent the biology of aging and create this new history. And this is a picture of Nico and me, that we were babies at the same time, and yeah, we don't want to go on talking like that and being like that. So what we're going to have here is 115 years is considered statistically, somebody said 120, the paper was 115 years statistically, maximum lifespan of human species. Now, I'm not saying that it cannot be broken, okay? But that's kind of where we are now with our technology. And we die, you know, 30, 40 years earlier, right? So I'm going to take the part of how we can get as close to 115 as possible. And Vittorio is going to take the part of maybe how we can pass that. I don't want to put words in your mouths, but just as a concept. And of course, we all discuss about the fact that, yeah, aging has a biology. That this biology is actually what drives aging, okay? You can be born with ApoE4 genotype, you can be homozygous, which means you'll be maybe demented when 60, 70 and dead at 80. But you're not demented when you're born or when you're 1 year or 10 year or 50 year, okay? It takes the biology of aging, and I can go through every one, and some of us did, through every one of the diseases and told you, no, aging is the major risk. Aging is what drives all those diseases, so we have to prevent them, and we saw examples that we can actually do that. And we can target aging, and we can delay, and in some places even stops and reverse some things. And I didn't want to just to forget to say that, yeah, maximize the exercise, diet, sleep, and social connectivity is really what we can all start doing right now. So, if you think of it, what we're trying to do, by the way, we're trying to really to focus on health span, but it's the same mechanism, okay? I think if we target health span, we'll have longevity. Sometimes I'm saying it's a side effect, okay? Maybe you don't like it, maybe you cannot afford it, but it's the same mechanism. And so we want to take a drug, okay, in a clinical study and show in maybe a 5-year study that we decrease mortality, okay, because it's not, we're not going to look at lifespan, okay? There's no experiment that will watch you for 40 years and then decide what to do. But in 5 years, we can see if we decrease mortality and if we decrease some diseases, okay, that are the equivalent of what we're trying to do with health. This is our goal. So the question is, how do develop text, or how do we look at existing drugs? And in order to understand what I'm saying, I just want to remind you about those hallmarks of aging. And I know it's a different slide. It's on purpose because this slide depicts the interaction between the hallmarks of aging. Okay? If I'll just take as example what Anna Maria did with protostasis, okay? And how do you become a hallmark of aging? You have to show that it goes wrong when you're old, that you can induce it in young, right? Like with the autophagy, that you can correct it by genetic ways, by drugs, and you can show that it extends health span and lifespan. And this is only by targeting, protostasis. And you saw other examples of targeting any one of those that did the same. And so I don't like when we all have those favorite hallmarks. I'm trying not to put anything in the middle. Just to know that any one of those can have really important results and affect health span. We don't have to do it all together to get an effect. So are there drugs that target the hallmark of aging? Yes, most of the drugs that we were talking about are targeting the hallmarks of aging. So for example, metformin does something, okay, that takes an old cell or an old organ or an old body and make it younger and targets all the hallmark of aging. So we have a lot of evidence for that. But metformin also in clinical studies and in association studies shows to prevent diabetes, heart disease, cancers, cognitive decline, Alzheimer's disease, and prevent overall mortality. So we have a lot of those data. How do we move to clinic? Just two things I wanted to say. I lost the vision here. Metformin in the 1920s, 1950s was an anti-aging drug. It was used for osteoarthritis. It was there to prevent flu. It was then that it was noticed that it lowers glucose and it became an anti-diabetic drug. Okay, but it was never starting as an anti-diabetic drug. It is not really the main point of metformin here. The second thing that is really important is for you to know that around the world, during COVID, 9 papers shows that people who were on metformin had half of the mortality, half of the hospitalization. And that led to a clinical study that showed that, sorry, let me see if I have this. No, I didn't have it there. A clinical studies that showed that if you give metformin within maximum 3 days of COVID, mostly 1 day of COVID, if you take metformin to people who are COVID positive, it delays hospitalization, mortality by 40% and long COVID by 60%. What I'm saying here is, you think metformin is diabetes? No, it's not. It's also targeting the immune function. Okay, it shows you that those drugs are really important for the hallmarks of aging. And that's what metformin has shown, while showing that it's clinically approved. But, and this is the point, we looked, I'm talking about a committee, I see the authors are not there, but they were basic science and clinical science. We published a paper 2 years ago and just last week, a follow-up. We took 12 drugs that all of them, when they're giving to animals, they extend lifespan of animals. Okay, that's what we took. FDA-approved drug that extend life in animal. It's very important. FDA-approved drug, which means they have safety, efficacy. People are using them. Okay, so we took them and have a scale of 12 to see if it's reasonable to use them in humans. And the 12 are 6 points are preclinical, hallmarks of aging, health span, and longevity. And 6 points are from human studies that include health span. And when I'm saying health span, if they were designed for diabetes, for example, but they actually prevented heart disease or kidney disease or liver disease, and also affecting not the mortality that is associated with the disease that they were targeting, but overall mortality, okay? And you see quite strikingly that there are 4 of them that get over 10 and over. And they are the SGLT2 inhibitors that get 12, the metformin, biphosphonates that were not on our scan before I read it first like 3 years ago that women who are treating for biphosphonate, by the way, it's anti-osteoporotic drug, that women that are treated with it are not dying if they're in ICU. You know, those women have not been dying. And then there are clinical studies, and they all showed that it treats other diseases and that it prevents overall mortality. And when you give it to animals, they live longer. Okay? And GLP-1 receptors, the ones that were developed for diabetes, Ozempic, right? They were developed for diabetes. They've been repurposed for obesity. And in people with obesity, they showed to prevent both cardiovascular disease and overall mortality. Those drugs have passed tests that are like the Tame trial. Okay? They have kind of different outcomes. They were not designed to show the outcomes, but they designed the same way, and they capture the same outcomes. And all of them are showing, all those are showing that they're that are effective. So our problem is that FDA doesn't approve aging as a disease. And if that's the case, healthcare provider will not give the drug. And if they don't give the drug, the pharmaceuticals will not develop better drugs and additional drug. And this is why we're doing the Tame study. But again, all the drugs that I showed you have safety, have efficacy. Metformin has been used for 80 years by billion years of people. We know everything that we need to know about it. Now, we always thought that supplements should come first. But we know now that supplements have huge problems and no clinical trials. We don't know if there's supplements in the bottle. If there's in the bottle, we don't know what it's doing. There's a lot of hope and very little promise. Doesn't it make more sense for us to go first to FDA-approved drugs that already have showed in big clinical trials that are preventing other disease, that are preventing mortality, and start thinking how to use it? And I'm not suggesting to use it now because maybe you need to have a little bit personalized approach to how to do it, but couldn't you do it now? Metformin, for example, has been repurposed for obesity. It's not the best drug, but people lose 5 to 8 pounds on that. PCOS, okay, young women with PCOS. COVID-19, I just told you. And pre-diabetic, there's the diabetic trial. Those are not FDA indications. But all the doctors are repurposing. If you're a doctor, okay, nobody is going to sue you for repurposing an FDA-approved drug. Now, it's difficult even for me to say what I just said, because the first day in medical school is primum non nocere, right? Is do no harm. Okay, that's what we learn. Think about it. If that's what you teach students, you do a curve, half of them don't want to give you anything. Okay? All drugs have trade-offs. You have a headache, they won't give you paracetamol because in 1 in 100,000 there's a liver problem, okay? The second day of medical school is there's no always and never in medicine. So we're conservative and we also are confused, okay? And I'm saying those are FDA drugs that have had all the things that we wanted to show. We should be able to use them. You can see more in our study, in our thing of the examples, the actual examples of, you know, effect size that are 20-30% over 3.6 to 5 years of what they're doing. It's not hard to understand it. So that's why we're doing the Tame study, and we want to do 3,000 people between 65 and 79 years old and have an outcome of a complex of diseases that we're trying to prevent. By the way, the kind of cool thing and what was part of the discussion with the FDA that are okay with it, by the way, if that's what you're going to show, we're going to be happy about it, is that we're kind of agnostic to what disease you're going to take. From us, I don't know what disease you're going to get next, but we'll prevent it because we're going to target aging. So if your mother is obese and you're If your mother was diabetic and you're obese, you probably get diabetes first. But for everything you get, you're getting point over time. Okay? And we'll show this construct, this complex of diseases is going to be prevented. Okay, okay. So this is my point and most important point. I think the future is right here now. We have to start explaining to people that from this perspective, we have geroscience proven drugs and we should think of using it. But can we do better? So the second point I want to make, and I'll make it short, is we have so those are 4 people that were born in New York between 1910 and 1920. And what's unique about them is all of them died beyond the age of 102. In fact, the young sister on the left died first at 102, and they were like shocked that the young sister died. The others died at 106 and 109, and the sister that is on the right, Helen, she died at 110. I saw her first when she was 100 years old in a New York apartment. She opens the door and she's smoking. And I said, Helen, nobody told you to stop smoking? She said, all 4 doctors that told me to stop smoking, they died. And actually, until 110, I was like, maybe you live longer if you don't smoke. And she says, when you smoke for 90 years, you live a long time. But really, I'm saying that, it's the truth, but I'm saying it because 60% of the men, 30% of the women were smoking, 50% were overweight or obese, less than 50% did exercise even as housework or walking. So in this group, it's not about the environment, okay? They ate what they shouldn't eat, vegetarian were 2%. So they are not the environment. We wrote extensively about it with control group. So it was something else. So we have 2 studies at Einstein. You'll hear more from Sophia Millman tomorrow about the offspring of centenarians that are much healthier also, and the other longevity study that includes centenarians. So we have centenarians and their offspring, and the people who are married to their offspring, so we can see how the longevity genes affect the offspring and how the offspring are healthier than their spouse, okay? And so you'll hear more about it tomorrow. But this is the important thing, 2 important things that I want to say. First of all, we always worried, are centenarians getting sick when everybody gets sick, when they're 60 years old and they're just sick for 40 years? Or is their health span and lifespan, does it go together? And this is a combination of 2 studies. This is from New England Centenarian Study. Tom Pearls ran this study. So they are black and red, and we are green and blue. And everybody is healthy. After the age of 60, a lot of them are getting sick. Only 10% are not sick after the age of 80. But you see that in each group, the centenarians live 20, 30 years more healthier. And it's an underestimate because remember when the centenarians were born, the people that were born with them died when they were 50 years old, okay? We're comparing the health span to generation below them. So they live healthy, okay? And basically they die. You see that in our population, 30% of centenarians don't have any disease. Many of them just don't wake up one morning. But this is not the important thing. The important thing is they have contraction of morbidity. So they're compression of morbidity, we're saying. They're sick very little time at the end of their life. We're sick 5, 8 years, and they're sick few weeks or few months, or they're not sick at all. So for me, this is what we're trying to achieve. We're trying to achieve, being healthy, and die. And it's not going to be so soon either. Michael told you about the longevity dividend. I'll just tell you that the CDC, since 1993, is showing the same data, that the last 2 years, the medical expense in the last 2 years of those that are 100 years old is 1/3 of those who die at their 70. Okay, so you can see that it actually like that. And I'll tell you, those 100 years old, when they were 60, 70, they didn't even go to the doctor. Okay, so there is a real longevity dividend by studying those people where it's not the environment, and they don't have, by the way, the perfect genome either, but something slows their aging process. And we found several important pathway. I'll just give you a taste, because those 3 examples have all been implemented clinically. One of them, the blue line, this is a certain genotype, and in those cases there are functional genotype. In other words, this change of DNA that has actually a functional consequence. This genotype is in the population about 8%, and it's more than double, okay, at 100 years old. So 100 years old have a lot of this genotype, which suggests that helped keep them alive. And this genotype is called CETP. It lowers CETP level, they have high HDL, they have a lot of clinical things that I'm not going to go through. But there are also people with that are protected from a variety of other age-related diseases. There is another genotype that's called ApoC3. It's also in the lipid pathway. It also is associated with less of this protein, a favorite lipid profile. And by the way, both of them have been validated. By the way, the CTP has been validated in North Italy. Okay, in North Italy, it's also a very important longevity gene. And the third one is a TSH receptor mutation. It also has been validated. But really the reason I'm bringing those, because there are some other things, but the reason we're bringing those is that those 2 companies have taken them, they underwent drug development. Okay, the fact that we have functional genetic mutation doesn't mean we have genetic therapy, okay? Most of the things are actually translated to drugs. And 2/3 of the drugs that were FDA approved last year were based on genetic data from humans, which really made it much more relevant, because when we thought that in mice we understand everything and we tried to develop drug, it didn't work. Okay, we need the genetic evidence. We have the genetic evidence here. Merck have produced the CETP inhibitor, Ionis, the ApoC3 inhibitors, and both of them are coming in drug forms soon. Okay, those are developed based on centenarians. The mutation in the TSH led to a change in medical practice, because if you know the screening for thyroid disease is checking your TSH level, that's the thyroid stimulating hormone, and if it's a little bit high, your endocrinologist might start thyroid hormone. And the point here is those people have high TSH, which is probably normal for them. And that led to not giving elderly, not suppressing that, because maybe by that you're increasing metabolism and you're causing harm. Okay, so I'm just telling you, we find an example of 3 genotypes. The 2 of them have been translated to medicine, 1 to medical practice, and that's why, from our perspective, the centenarians is where you want to find those genes. American Federation of Aging has a super ager initiative that recruits 10,000 more families that will have, you know, the centenarians, a child and somebody who's married to the child, in order to validate and find more longevity genes, because those probably are going to be the important thing that slows aging. Tomorrow you'll hear more about growth hormones, because I think growth hormones are one of the major modulation of aging. You have to have less. Sophia Millman will talk about it. And so could our kids, you know, Nico, could you be a healthy centenarians? So first of all, get ready for those gerotherapeutics. I want to say that it's not only about older adults. Okay, people who survive cancer because of radiation and chemotherapy are aging quickly. People with HIV are aging quickly. People who are disabled. Poor people around the world. And if we want to go to Mars, we have to stop aging first, because we'll never get there without cancer, cardiovascular disease will never make it back. So that's it. We have to spend our time on health and not on diseases, and let's see how we go on. Thank you. Not too bad.
Nir, thank you so much. The other day I asked you why you do what you do, and you told me that it was for me, and not just me specifically, but I think when he said it was for me, he meant everybody in the world, everybody, all of you here. And I just want to say, every time I speak to you, I'm more hopeful about my own health and also just the future of humanity. So thank you. You can tell that this is this research comes from your heart, and I really appreciate it, and so does the younger generation. Next, I would like to introduce Vittorio Sebastiano. This is his hometown, so I'm thrilled that he's here. You're thrilled that you're here, and I'm sure the audience is thrilled that you're here. He is a trailblazer in the field of regenerative medicine and stem cell biology at Stanford University. His groundbreaking research on cellular rejuvenation and stem cell therapies has earned him prestigious awards, including the AFAR Junior Investigator Award and the Glenn Breakthrough in Gerontology Award. He gives off a little bit of mad scientist vibes, but I assure you he's very sane, and his research is very exciting. So I'm excited to hear about it. Thank you.
Just paint. Oh, it works. Well, good afternoon, everybody. It's really a pleasure to be here today. Yeah, this is my hometown. I grew up here, and it's really a privilege to be here and to share the stage actually with Nir, a scientist I deeply, deeply respect. Let me say, first of all, thank you to Viviana and all the organizers. You have done an incredible job in putting this together, and it's really a terrific event. So I'm really, really pleased to be here. So today I will share, in the next 20 minutes, I will share a little bit about how I think we could envision the future and the future of anti-aging medicine. And I want to start by actually saying that as I was preparing this talk, I found this image on Google, which is really evocative of what I'm going to tell you in the next 15 minutes or so. Evocative of the fact that I believe that the future, or at least some of the secrets for developing anti-aging solutions really are in the egg, in the oocyte, in reproduction, okay? So this morning we had a very fun chat with Nicola Marina and others, and we were saying that, I'm going to say this in Italian, that la longevità è donna, longevity is woman, I guess, this is the translation. And it's woman for two reasons. Well, first of all, because we heard from Camilla before that there is definitely a better job we have to do in understanding why women age so differently from men. And she said it's unfair. I share that sentiment, that the aging process is so sexually dimorphic in an unfair way. But also because I think longevity is woman because the future, the secrets for anti-aging is in the biology of women. Okay, so here. Okay, so well, first of all, let's say why do we care about all of this? Why all these people? So so many brilliant people are here, gathered together, from investors to scientists to clinicians. Well, the reason is very obvious. It's obvious because getting older means being at risk of developing one or more of the conditions that you see here. And if you combine that with the fact that we are aging very fast, and in about 30 years from now, about 20% of the population is going to be age 60 or over, that means that there's going to be 2 billion people that are going to be at risk of developing one or more of these conditions, because these conditions are caused by the fact that people age. Aging is the #1 risk factor for all of this. And so the question is, if we can stall or even reverse the process of aging, maybe we can, start thinking about a way to holistically and comprehensively maybe try to tackle all of this all together. And so how historically the approach that we have taken has been preventative, in the sense we have heard it from many speakers before. We have been trying to fix one piece of the puzzle at a time, and by doing so we have definitely done a good job in extending the lifespan, but at the same time we have increased the window of time at which frailty kicks in. And so we are exposed to frailty for a much longer time. More recently, we have been starting thinking about a different way, intervention. So can we stall the process of aging at the cellular level? Or can we even reverse it so that now we're going to live longer lives, yes, but we're also going to live healthier lives. And then we're going to have that sudden death that was mentioned before. So we're going to die young, in a way. So how do we tackle this problem? Well, I believe that one of the most fundamental reason why we age is in the what I call up there the epigenetic program that becomes an epigenetic problem with time. So our cells have, as you know, a genetic code and an epigenetic code. The genetic code is the one that we inherit from our parents, and for the most part is fixed. It's not completely true, but let's go with that simplification. But the epigenetic code sits on top of it, and it's the reason why genetically identical cells in your body are so different from one each other. And that's the reason why cells in your brain are so different from the cells in your liver or the cells in your pancreas, and so on and so forth. And that's because the epigenetic program tells what genes are supposed to be on and what genes are supposed to be off. Now with time, as you can see here, with time that program becomes dysfunctional. And by doing so, it affects a variety of different things in the cells, the so-called hallmarks of aging. And so that's the hallmarks of aging are the manifestations of aging. But I strongly believe, and I'm not alone, that actually it's primarily because of the epigenetic changes that are both developmentally regulated, meaning that there is a program in our cells that allows that to happen. For example, lack of maintenance, as we heard before. And then, of course, there is an environmental factor that is additive on top of that. But it doesn't matter if it's really the root cause of the aging. What really matters is that the epigenetic code is reprogrammable. And so here we're thinking about the possibility that if we can take that dysfunctional program and we can revert back to a functional program, then by doing so, maybe we have an opportunity altogether to affect most, if not all, of the manifestations of aging at the cellular level. So it would be a more comprehensive way of looking at rejuvenation, if you wish. Great. So how do we do that? It seems like a very challenging task. Well, I don't think we have to reinvent the wheel. We have to. What's going on? Okay, we don't have to reinvent the wheel. I think that we really need to kind of look at nature, because nature has already figured out a very elegant way to do this. And that's in reproduction. That's in embryonic development, in germ cells developments, and of course, as I said before, in women's biology. Why? Think about it for a second. We do age, right? We see that happening every day in front of the mirror, right? So every single one of us is aging as we speak. But the human species as a whole is not aging over time. Every time we have babies, the new generation is born at age 0. So there is no vertical transmission of aging across generations. So that means that there is really something special about embryonic development. There is really something special about reproduction that allows for that glimpse of aging that obviously occurs even in germ cells to be completely erased and reset to 0, so that the baby now is born age 0. So we just have to look at that. We just have to understand that process, and we have to take it and apply it to the soma, to the body, to the cells that are non-germ cells, but are the cells that make up our body. And so, well, is this science fiction? No, it's not science fiction, because there is already plenty of evidence in the literature that this is conceptually possible. So think about the groundbreaking work of Shinya Yamanaka, who got the Nobel Prize for discovering a way to make induced pluripotent stem cells. So these are cells, as you can see here, that can be derived in principle from any cell in the body. So you can take an old skin cell, and by reprogramming it, you can make a young embryonic-like cell that now has this incredible potential to differentiate into any cell of the body. And so this is great because, you know, we can use it. You can use these cells for regenerative medicine to make organs, right? But there's something more into that, because the cell now is not just embryonic, but is also very youthful. To the point that now if you take that cell and you differentiate it into the original cell type, let's say a skin cell, the skin cell that you make now is a skin cell, but is also a very youthful skin cell. So there's two things going on during reprogramming to iPSCs. There is rejuvenation, massive rejuvenation, and there is also change in cell identity, meaning that a skin cell becomes an embryonic cell, right? And so in order to make a new skin cell, now you have to redifferentiate it into a skin cell, but that's not easy. So what about skipping the cell identity change problem and just take advantage again of the fact that the cells are also becoming younger, right? What if we can just make that skin cell a younger skin cell, but not having losing its identity? This is called partial reprogramming, or transient reprogramming, or interrupted reprogramming. I call it ERA, epigenetic reprogramming of aging, but it doesn't matter. It's the same concept. And so when I started thinking about this problem, you know, you can tell that I'm not a mathematician, so my equations are very, very simple, unlike the equations that we saw before. So at the beginning, when I started thinking about this problem, what I came up with was a very simple idea that maybe the reprogramming or the rejuvenation aspect is just a function of time, of expression of the reprogramming factors that you put into the cells to make iPSCs, and a function of the dosage, meaning the amount of expression of those proteins, as you can tell here. So Y is the degree of cellular rejuvenation, and it is a function. It means that it's proportional somehow to how long the factors, the rejuvenating factors of the cells in the cells are expressed, and how much, in simple words. And so what we have shown is that you can really take an old dysfunctional cell that you see on the left, bring in some mRNAs, a cocktail of factors, and by doing so, expressing them for a very short time, as if it was an embryonic-like reprogramming, but just very short in time, you can make the cell younger, but the cell is still the same cell type. So there is retention of cell identity, okay? And I'm happy actually to see that, to say that, you know, together with the first seminal work of Alejandro Campo and Juan Carlos Itzpizua-Belmonte that you heard yesterday, our work that was the first in human cells, those were really like the two milestones actually to really establish a new field, which is this transient reprogramming field that now, as you can tell, is really exploding. And tells us a very important concept. Well, first of all, you know, this is reproducible across labs in different animal species. So it's really a reproducible technology that can have a potential to be utilized in different contexts, because it doesn't work only in different animals, including humans, but also in many different tissues, okay? And so very quickly, I want to tell you how we did it. So, well, first of all, it's very important how you do this process. We chose the bottom, the mRNAs, because we think are the most safe approach to do this. And it also, most importantly, give us an opportunity to control a lot of processes that are very important, but most importantly, they can be made at scale, which means that this could potentially be a solution for democratization of this medicine, which is, again, not just for a few, it's for everybody, because this is a problem that everybody will experience in life. And so what's left to the cells is just translating that message that you're providing them and going through that transient reprogramming process. Does it work? Yes, it does. So here you see on the left, for example, you take fibroblasts, the dermal skin. So on the left, you see gene expression. When they are aged, they have a very low expression of some markers, a very high expression of other bad markers. When you transiently reprogram them, they start overexpressing the good genes, and they start downregulating the bad genes that you see here in blue and yellow. There is also an epigenetic rejuvenation, meaning that the cells are, from a methylation standpoint, younger. And this technology can also work in stem cells. Why is it important? Because stem cells are the cells that are responsible for the regeneration of tissue over time, over a long period of time. So in collaboration with Marco, who I believe is in the audience, I didn't see him yet, but what we did was to try this concept in muscle stem cells. And what we saw was really remarkable. We took the muscle stem cells from a mouse, we reprogrammed them for just 2 days in vitro, and then we transplanted them back into an age-matched mouse that was also injected with a cardiotoxin. It's a molecule that basically destroys the muscle. So the cells were challenged to regenerate the muscle as they were after injection. And what we saw was that we could restore the regeneration capacity of young, untouched muscles. That is about a 40% of induction of regeneration and induction of force. If you want to translate, I mean, I don't think it's really fair, but if you want to translate, so we did this from mice that were 20 months old, and the muscle went back to the strength of a 6 months old. If you want to translate this into human years, we're talking about decades of rejuvenation, okay? The important thing to say is that, it doesn't just work in our hands. We are in the land of Galileo Galilei, so, the experimental method and the reproducibility of data is the most important thing. It's a technology that works, that is scalable. It works across different cell types. And I'm not just, I'm saying it for a fact because you can see that it has been tested in many different animals, in many different tissues, using a variety of different strategies, and it does work across different tissues and animal models, and so it's really a reliable method that could potentially revolutionize the field. Not only that, it's also very important because you can use it as a discovery platform, for example, identifying new genes that are involved in this process. You can also utilize it to, for example, to maybe not systemically rejuvenate, meaning that you go into every single cell of the body. No. As we understand aging, and maybe as we understand that maybe there are some organs that are the major drivers of aging systemically, maybe we could go into those organs, rejuvenate those organs specifically, and by doing so, we could rejuvenate the body. And for example, the ovary could be the first target for such an approach. But then, it can also be applied to something that has nothing to do with aging, chronological aging as we mean it. There is a lot of conditions that are aging-like in the sense, for example, obesity, for example, post-traumatic osteoarthritis. So that there's some phenomena, inflammation, damage, that induce an aging-like phenomenon in the body that has the same outcome of chronological aging. And so you can intervene in those conditions, and with the same idea, you rejuvenate the cells and you make them younger again. Or in other potential applications, for example, CAR-T exhaustion. So with that, I'm just a little bit over time. I just want to thank the people in my lab. I want really to thank everybody in the field, the people in my lab, the funding, and yeah, thank you.
Will you stay up here? Okay. Thank you. Thank you, Vittorio. Really really appreciate your talk. Now we're going to have Vittorio and Nir come up for a short discussion. I have a question for you, and unfortunately we cannot take questions from the audience now because we're a little bit over time, and we have a hard stop at 6:30, but maybe I'll ask a question and then you guys can have a short discussion. Is that okay? Sure. Okay, amazing. Wow, I can't believe I'm sitting up here. So thank you both. This is something that's been at the forefront of my mind. And Nir, I asked you about this in the past. I got a feeling you didn't want me to ask you about it, but I feel like I have to as part of the younger generation here. The thing that scares me the most about aging is the amount of foreign bodies like pesticides and hormones and heavy metals that are making their way into our food, into the water that we drink. And I know for a fact that many young people are, simply because we have to be, because we're seeing the effects of it. And I know that you deal with the biology and the genetics of aging, both of you, but since we are seeing the hallmarks of aging that you described earlier come at a younger and younger age for many people, how can you really ignore the environmental factors? And I'm not saying that you do. This is kind of a theoretical question.
No. First of all, you're right to be worried, okay? There's no question. If I gave you the impression, don't be worried, I didn't. But I just want to pick on Michael Ringel. You know, if we save $360 trillion in economical value, we can deal then with the environment by spending $1 trillion, right? Or something like that. But look, I think the point is that in the last 150 years, you know, this longevity thing, right? I mean, you know, COVID did decline a little bit, but this is, so in spite of the environment, we have actually made progress in our health and our longevity, okay? So I would be optimistic, but I wouldn't say that you're not. You know, look, COVID, maybe it's not what you were talking about, but COVID is an environmental, you know, it's because of environmental something, right? And so you're absolutely right to be worried. We're worried all the time. And I think the more geroprotective we'll be, the less we'll let it, you know, what is more important? You know, people are asking in aging, what's more important, the genes or the environment? And the truth is that it's interaction, okay? It's interaction between the gene and the environment. If we could manipulate all the genes that are important, we can prevent the effect of the environment, okay? So I think you're right, but we're on the right track because of that.
I agree, and thank you.
No, I absolutely, I agree as well, you know, with what Nir said. Maybe, I guess, maybe we can take that, I can take that as an opportunity actually to say something else, in the sense that let's not think about what, you know, we are doing as, oh, one day there's going to be a pill that is gonna, you know, prevent aging from happening, and so we can do what the hell we want, you know, and then we take the magic pill and we go back in time. I mean, well, I think that we should all take a very proactive approach, and we have heard this, you know, many times. We can do a lot about aging, you know, as we speak, you know, exercising, having a good diet, and so on. The society can do a lot about aging, because it doesn't matter how long we live, if those lives are going to be, you know, miserable and you know, alone. I mean, there's a lot that we can collectively do, including, for example, making sure that maybe, you know, we should start stopping using pesticides and things like that. So I think this is an opportunity really to think as a society, as a group, about the fact that, well, let's not just do this in the hope that there's going to be a magic solution for, you know, whatever lifestyle we want to have. It's an opportunity to develop technology, but also to think as a society that there is a lot to do about the problem of aging, because this is really a problem, you know, at all levels: societal, anthropological, financial, medical. So there's a lot that we need to do together.
So can I ask him something? Right, we're asking each other.
Yeah, you guys are.
I'm saying a sentence, and please say if it's yes or no. In 50 years, a 20-year-old, let's call him Peter Pan, but doesn't matter, will come to a doctor, will get a treatment, maybe injection, that will be followed every few months or every few years, and they'll stay relatively young. Yes or no?
I don't know if it's in 50 years, but yes.
What would be your guess? By the way, 50 years is because I don't need to be responsible for my answer then.
I think it's a foreseeable future. It's at grasp, honestly. And this is, by the way, not only, it's going to be a combination of different technologies together. It's going to be organ regeneration, it's going to be senolytics, it's going to be partial reprogramming, it's going to be metformin, hopefully, and a lot of different things together are going to get us there. Absolutely, I truly believe it.
Now I'm quoting you, so it's okay.
Are there any other topics? I was sorry, guys. I was prepared to take questions from the audience, but we had a little bit of a curveball.
That's Viviana says. Why don't you go on in? So can I ask you another thing? This is right. When I said the hallmark of aging, I said I didn't say mechanism of aging, and I kind of said I don't like when people are putting their thing in the middle, right? And you put the epigenetic in the middle. So I will ask you that. So when you're old, you can see epigenetic effect. You get hair where you don't want to get. You get fat cells where you don't want to get. And you're saying there it is, epigenetic is. But what causes this epigenetic malformation? There's some other mechanism than epigenetic. And I would ask everyone, if you use mTOR, right? So mTOR is high, you want to bring it down. But why mTOR is high?
That's why I said, I'm not necessarily fixated about the idea that it's the cause of aging, but it's one of the most fundamental way by which we could intervene on many hallmarks at the same time by working on one mechanism, okay? I'm always kind of, struck by the concept that when we are young, we are actually subjected to a much higher degree of damage than when we are older because we have an incredibly unhealthy lifestyle, right? But still we can cope, we can cope with that, right? We can cope very well with that. So how is it possible? If the theory of the damage, and I'm not saying it is not true, but if the theory of the damage was the only theory, we should, have an accelerated aging, very fast. But that's not happening. We can fix that damage very efficiently. But that capacity diminishes with time. So, and I think that most of it is because there is a lack of maintenance of mechanisms responsible for that, which are in the genes, which are in the epigenetic program, and that's why I hope that could be a very deep way to fix the problem.
To interrupt you, but the theater is telling me that we have to close. They throw us out, they have an evening show here, and so we promise we leave at 6 and we have to go. But maybe, Antonella, you wanted to make a fast question.
And good evening, everyone. I will have 2 questions for those distinguished colleagues.
One.
The first one, it is, we know that women do age worse, although they live longer. Based on your expertise, how do you envision, you touched very briefly on using this approach to rejuvenate ovary. Can you just explain us a bit more, and do you think it is sustainable? And the other quick question, have you thought of using all the amazing drugs that are repurposed and already approved by FDA in something like progeria, like an orphan indication to disrupt the way FDA might approve those drugs?
Well, the answer for the second is yes.
Good, so very fast. Fantastic.
Okay, the answer to the first question is that I think that the aging of the ovary is going to give us, the rejuvenation of the ovary is going to give us a tremendous opportunity to do 2 things. On the first hand, to try to tackle the concept or challenge the idea that women shouldn't be having a child later in life, because rejuvenating an ovary means rejuvenating the follicles, and so the ability of a woman to have children and not being put in the condition of choosing to have a children early in life, because otherwise the biological clock clicks. The second thing is that the ovary has a clear role in the systemic aging of the woman, And so by rejuvenating the ovary, you potentially have also an opportunity to systemically impact the aging process. So it's one example where targeting one organ could give you a lot of different outcomes.
Thank you.
Thank you very much. This was, 1 second, don't get up yet. So I think this was mind-blowing. I invite you to be here tomorrow, and if you're interested, I can tell you what is my recipe for longevity. And my recipe for longevity is this young man. Nico, come here. Nico is my grandchild. I'm very proud of him. And I think the best longevity is the legacy we can give to our children and grandchildren, so they bring whatever we have. We cannot transmit culture, but we can transmit the thirst of knowledge and the pleasure we have in knowledge to them.
Very well said, Vivi. Vivi.